Right. Like I said, it's pretty low key, so.
Yeah, and that's what like I've been told. Yeah, it doesn't. You know the stuff. You know the stuff better than anybody else, and then
You're the expert.
if you don't know, just say that. Oh, that's a great topic in Chicago. We should look into.
Yeah, they're always there.
Yeah.
Yo. not sure what, I'm not sure what happened. But please feel free to get snack or drink, there's coffee back there as well, and we're gonna wait just about 20 more minutes. Thank you very much for your patience. Morning, everyone. We're going to go ahead and get started. Thank you for your patience. I appreciate that. So welcome to the Regional Science Consortium's 20th Annual Research Symposium. This is an exciting year for us. It's 20 years. I'm Jeanette Schneider. I'm the Executive Director of the RSC. And the Regional Science Consortium is located in the research wing of this building. So on the other side of the building, we have several laboratories back there that we do our work. So this year for the symposium, we have a lot of presentations, more than we've had in years past. So we have 43 oral presentations, 58 poster presentations, and 20 art exhibits. And all are given by undergraduate and graduate students, researchers, artists, and faculty. In addition to the in-person, all of the presentations will be live streamed, thanks to Eric over there making that happen. And so if over the course of today or tomorrow or the next day, you're unable to come back here and watch the presentations, log into our YouTube channel at RegSciTV.com. I also wanted to mention tonight is our poster session and art exhibit, and that is from 6 to 8 p.m. Like I mentioned, we have 58 posters and then 20 art exhibits, and we also have refreshments, so please come back and check that out. It's a fun time. All right, so if you need anything over the course of today, please stop at the registration table. After this session, we'll have lunch. All are welcome and invited, so please join us for lunch after this session. And I'm pleased to start this year's symposium. All right, we are going to start with Andrew and Matthew from Gannon University, and they are going to present on the pharmaceutical intervention of the obesity crisis. And you guys want to come on up? Thank you for kicking off our symposium. The middle button is right there.
Oh,
Forward and
we
backward.
brought our own.
Wow.
Awesome. Well, thank you all for being here. Hello and good morning. Hope everyone's having a great start to their day, even despite this wonderful, eerie weather. So I just wanted to start off by introducing myself as well as my partner. My name is Matthew Nicolo, and this is Mr. Andrew Burton. We're both biology students at Gannon University on the pre-health track. I also wanted to say thank you to you all for the RSC in providing this wonderful symposium to allow us to share our research, as well as to our professors, Dr. Prasad Dalvi and Dr. Liu. Thank you for guiding us along with this research over the past few months. So, let's get started. Our presentation today is going to be on the pharmaceutical intervention of the obesity crisis from origins to innovation. Obesity is currently a global health crisis that demands for more effective and safe drugs. This morning, we're going to be diving into the complex factors that are contributing to this obesity crisis, as well as looking at the historical origins all the way up to the future of these newly developed GLP-1 agonists. So Mr. Andrew Burton is going to start us off with giving us a bit of background information on what obesity actually is, as well as viewing some current trends and patterns that we're seeing across the globe. Andrew?
Thank you for that wonderful introduction, Matthew. As you said, my name is Andrew, and I'm going to give you guys the statistics and background of obesity. Obesity is a worldwide phenomenon and is currently affecting the entire globe. So we coined this nice, catchy little term, globesity. So to start, Obesity is characterized as an excess accumulation of body fat, and this severely increases one's chances for adverse health outcomes. MRI and DEXA can be used for precise measurements, but more common and practical methods such as BMI and waist circumference can be done at home and are much more cost efficient. BMI stands for body mass index and is a ratio of one's weight to height. A typical BMI is anywhere from 18 to 30 and varies upon metabolic rates and stature. Typically for Asians, theirs is around 24 due to environmental challenges and different varying environmental issues, I'd say. Waist circumference. For males, typically the waist circumference is anywhere from 80 centimeters and less, and for females, 90 centimeters and less. Prevalence and impact. As I said, obesity is currently affecting the entire world, with approximately 1.3 billion people affected worldwide. Certain trends could be noted, such as different socioeconomic, racial, and gender groups being disproportionately affected by this trend. This strains the healthcare systems worldwide. And there are certain key contributors that we can look at, such as family dynamics, socioeconomic divisions, declining food quality.
Sorry.
It's all right. So, sorry for that distraction, but let's look at these graphs here. So these are showing the exponential increase in unhealthy weights. So the top two graphs is male versus female obesity worldwide, and boys versus girls. So we have children on the bottom and adults on the top. If we look here at 1980, we can see that this little extrapolation of data here is significantly less than if we fast forward to 2010 in the present, with approximately 300 females worldwide, 300 million females worldwide being affected by this. This is almost a 10-fold increase from the 1980s. So now if we move forward, let's look at this heat map. We can now further dive into the different disparities in the world. So the highest rates of obesity are in Eastern European countries, with the entire Eastern European population having approximately 60% being afflicted with obesity, with 54% being females and 46% being males, and 4.5 million children in Europe being affected with obesity under the age of 5. So let's also look at some different global trends. There are 1 in 3 men affected with 1 in 4 women affected. If we look at South Africa down here, South Africa, there is a significant prevalence of females who have been diagnosed with obesity versus males. And there is a trend. Obesity is correlated. There are higher rates of obesity in lower educated communities. And there's also higher rates of obesity in higher, in bringing in countries like higher GOP companies. So BMI, as I had said, is a way to measure one's health. For every 5-unit increase in BMI over 25, this can be equated to a 210% increase in diabetes-related mortality. So now that we understand how significant obesity is, let's look at some of the key contributors. So, socioeconomic disparities. As mentioned, with the disproportionate events in this world today, there are many different socioeconomic classes, with those in the lower ones being significantly more affected. Coupled with sedentary lifestyles, this can increase one's rate for obesity. Sedentary lifestyle is any lifestyle that is that of decreased physical activity, and that can be for someone who's going from a physical lifestyle change to a desk job. So let me ask you, do you know how many hours a day do you sit? If not, that's okay. Typically it's more than we would like, and that's all right. So that's why we're addressing this issue. Family dynamics. With the increase in constant stress and increase of single-parented families, this negatively impacts one's eating habits and activity levels. And the most important factor, the declining food quality. This is currently affecting all of us right now. Let me ask you, have you ever looked at what you're eating, what's inside, what you're eating, and what's on the nutritional label? If not, that's all right, we're going to take a look at it later. So this is affecting us all, as I had said. So let me give you some basics. 1,500 to 3,000 calories is the normal range for a human of caloric intake, with men being anywhere from 1,500 to 3,000, and women being anywhere from 1,500 to 2,500. This varies with lifestyle, activity, and metabolic rates. Sugar is recommended to be 6 to 10% of your daily caloric intake, with that equating to 100 to 400 calories. So let's look. Sugar tends to have a bad rep, but not all sugar is necessarily bad. There are natural sugars and added sugars. Natural sugars come from vegetables, fruits, dairy, and wheat, and contain essential vitamins, minerals, and nutrients that come along with it. While added sugars are any sugars that are added in the manufacturing process to include sweetness into the product. Some of these examples are aspartame, sucralose, and high fructose corn syrup. So now I'd ask, have you ever looked at a nutritional label before? Do you know where you can locate the sugars in a nutritional label? You can locate them in the total carbohydrates. Sugar is a carbohydrate, and typically it can be divvied up into added versus natural. But the funny part is, you don't see natural. Most of all products just simply include added sugars. So let me kind of put this into perspective really quick. One singular can of Coke has 39 grams of sugar, and the daily recommended intake of sugar is 72 grams. So one single can of Coke has more than half of the daily required amount of sugar. But that's not all. If you start to add this up, one serving of Fruity Pebbles, 31 grams of sugar. One serving of Heinz ketchup, 5 grams. If you begin to add these up and don't pay attention, maybe you could look like this. Not really, but potentially. This is why we're doing our research here, is to prevent this. Food is significantly decreasing in quality, and people are becoming heavier than ever before. And we're trying to help prevent that. And some ways that this affects the human body is kidney cancer, the brain, liver, and especially type 2 diabetes. That is the most prevalent outcome of obesity so far. So before I hand it off to my partner Matthew to talk about the first pharmaceutical drugs to combat obesity, the missteps along the way, and the potential for GLP-1 receptor agonist drugs, I give you all the background information, so Matthew, please take it away.
Thank you, Andrew. So as Andrew explained, there is a massive increase. I know you guys all paid attention to those graphs, maybe not, but they were all massively increasing. Like it's pertinent that this is a problem and a crisis within the world currently. So now we're going to take a look at the historical perspective of these pharmaceutical interventions on these anti-obesity drugs. So let's start back down into the 1950s. All right, the 1950s, the 50s and 60s. This is when President Eisenhower and President Kennedy were in office. Something that, I don't know, this looks like a young group, but some of you guys might remember that they actually installed and created fitness programs. This specifically is because there was a rising prevalence and concern for obesity within the United States. So let's go back to 1950 when the first synthesis of anti-obesity drugs began. Amphetamines. Amphetamines was the first drug on the market to combat weight loss. This was done through the mechanism of action to suppress one's appetite. But it was quickly halted in the 70s and 80s, specifically because they saw a massive increase in addictions, as well as increased risk of cardiovascular issues. So, the only things that we see amphetamines being used for medically currently is for child ADHD, as well as the more well-known issue of illegal use. So now let's jump up about 40 or 50 years to the 90s, orlistat's debut. Orlistat took a rather different approach by inhibiting fat absorption and produced a moderate amount of weight loss. So, this is still being used today as it's a significant step within the right direction of these anti-obesity drugs within a historical perspective. Though, looking into the 20th century, there was a lot of lessons and setbacks that we have learned over the years during the so-called drug boom, such as drugs like fen-phen and sibutramine. These caused severe side effects that no one would want to experience. All right, so now we're going to take a current look at the anti-obesity drug market. The innovations within the field currently have increased significantly. So we're going to start on phentermine topiramate. Phentermine topiramate is related to amphetamines as it's within the same drug family, though it's still only producing a moderate amount of weight loss. Next as I mentioned before, orlistat. It takes a different mechanism of action and inhibits fat absorption, but is still being used today as it's very promising, though still only producing a modest amount of weight loss. Now we'll direct our eyes to the bottom, naltrexone bupropion. This takes a more unique mechanism of action and a rather different approach as it affects the brain reward system by balancing those natural brain chemicals and making you feel more full, thus suppressing one's appetite, and still only producing a moderate amount of weight loss. Now we can direct our eyes up to the top. Liraglutide. This is one of the newly developed GLP-1 agonists that have been created recently, similar to Ozempic as well as its higher dose form, Wegovy. These are all GLP-1 agonists. They take and regulate your appetite as well as glucose and have the highest efficacy in comparison to any other drugs on the market, as they produce a significant amount of weight loss. So one category that I failed to mention is side effects. Liraglutide and orlistat having gastrointestinal issues such as discomfort and disturbances, phentermine topiramate causing dry mouth and constipation, and naltrexone bupropion causing nausea and headache. So I think it's very important to note that if we scale out to a macro view of the entire drug market, more than half of the drugs that we see today have side effects, more than 10 side effects to be exact. So I think that's just important to note as we weigh the efficacy and that benefit-cost relationship on these drugs. So now we're going to take a further dive into the newly developed GLP-1 agonists as they reshape this obesity treatment as well as reform and innovate within these recent years. So if you're unfamiliar with, the human body and all the hormones that are being produced, there's a million, I know. But GLP-1 is actually a hormone. Its mechanism is when you digest food and it goes into your GI tract, GLP-1 is then produced and diffused out of your GI tract, targeting both the stomach and the liver. It then is able to help increase your insulin as well as decrease hepatic glucose output as that insulin is uptaking that glucose. Thus, it's going to slow your digestion by slowing and decreasing gastric emptying, making you feel more full, and in turn, suppress your appetite. So this GLP-1 agonist is a mimic of that GLP-1 hormone. So they have the exact same route and mechanism of action, just the GLP-1 agonist would be a drug that you consume, dissolve into your stomach, and then diffused out of the GI tract. So this research and data has been supported through clinical trials such as STEP and SURMOUNT, which have showcased its superior efficacy in comparison to these other anti-obesity drugs within the market today. Now, I think it's also very pertinent to note patient voices and their perspective, as they're the individuals taking the drugs. Patients are basically saying that they have an extreme effectiveness of using these GLP-1 agonists, as they have significantly shown that their quality of life has improved, though cost and side effects still do remain a concern, which we will touch a little more on in the following slide. And next up, the future directions, as this GLP-1 agonist research is rapidly expanding. They are now beginning to personalize and individualize these drugs specific to one's genes and therapy through a variation of gene therapies. So now we're going to look at the cost and complications of the previously mentioned drugs in order for your own opinion to weigh the cost and benefit of the efficacy. So we're going to start off with phentermine topiramate and naltrexone bupropion. As well, these are all prices on a 30-day supply, just for you to note. Phentermine topiramate and naltrexone bupropion are relatively low in cost, but cause significant side effects such as birth defects, hepatotoxicity, seizures, and even insomnia. That's I don't personally want to take those. So those still only provide a moderate amount of weight loss. Next up, we're going to go look at orlistat. It's been here since the 90s and it's proven its efficiency. It's relatively cheap, as those are two name brands varying in price for $60 and $100 to $200, and cause minimal side effects such as diarrhea, flatulence, as well as oily discharge, though it's still only producing that moderate amount of weight loss. And now we can take a look at liraglutide, which is one of those GLP agonists that I have mentioned. The price is significantly higher, $1,500 to $1,700 for a 30-day supply. And they have some complications such as nausea, elevated heart rate, hypoglycemia, as well as C-cell tumors. And the outcomes we see are an enhanced quality of life, lowering the blood sugar, such as treating that type 2 diabetes, as well as a significant amount of weight loss. I also think it's extremely important to note that as we see the side effects in comparison to the price, as well as modest, and a significant amount of weight loss, those prices to that efficacy is directly proportional. So now we're going to start to look at, that historical origin, those past ideas and drugs that have been synthesized, as well as the current market and what it looks like, in order to help develop that future and forward-looking perspective on the obesity crisis. So we have learned a lot of lessons about these drugs, specifically the safety over a long-term period and the treatment plans that have been developed. A lot of drugs are only looked at in a short window. So we're now able to look at the underlying causes of what actually is causing obesity. As many of you know, in order to solve the problem, you probably need to look at how the problem is being caused. So the new horizons, these are specifically those GLP-1 agonists. This is an entirely new and recently developed mechanism of action, and we have seen a lot of benefits so far from these drugs. The ethical considerations is just, adhering and looking at how the drugs have evolved across the entire board, not just anti-obesity treatment, but we now have much more safe protocols in order for a company or research company to reach those clinical trials, which is extremely important as safety is a top priority. Now I ask all of you, I don't know if you're going to do anything about it, but we would love to continue this research because in order to continue, there's been a massive increase in innovations, especially with this GLP-1 agonist, as too many mechanisms of action have been produced over the years, and now we've limited it only to a certain amount, and we need to continue to do so in order to ensure that all of the patients and people that are using these drugs are able to have a healthy lifestyle while taking it. And these GLP-1 agonists have represented a significant amount of innovation within the field of obesity. And we need to help combat this ongoing crisis as there is a global burden on all of us, as some of you may have seen and even witnessed. So looking to the future, we need to help collaborate together in order to continue this research. We need to promote these healthy lifestyles and active lifestyles, as well as creating new policies in order for this combated angle on obesity to be reduced. Because the best way to treat healthcare, the best way to solve healthcare is to treat sick care. So thank you all for listening to our presentation today. Hope you all enjoyed it. Are there any questions?
On one of the previous slides, you said that one of the GLP agonists, the liraglutide, can cause C-cell tumors.
Yes.
What are C-cell tumors exactly?
So specifically C-cell tumors they're actually a very rare form of a tumor. I don't know everything about them, but I can tell you that they're extremely rare within the population, and there are surgeries in order to remove them. They're not necessarily malignant, but yeah, it's kind of, yeah.
I got two questions. Go back on the tumors. When you're doing your research, do you see any, they sort of talk about the worrying scams they're starting to see with the MRIs, with doing some good usage nowadays, especially with how there is like a trend that is the benefit over like weighing like the short-term benefit overweighing the long-term benefit, and then so yeah.
Yeah, so I would have to say that your own opinion weighing that cost-benefit relationship in terms of those adverse side effects in comparison to the efficacy of those drugs specifically. Orlistat, it only produces, I believe the exact number is about 5% of weight loss is contributed towards that drug. And, it's with minor, diarrhea, flatulence, and oily discharge in comparison to the others, those are relatively mild. So it is about, that future research and that future plan in order to develop these GLP-1 agonists such as liraglutide, Ozempic, Wegovy, in order to lower those adverse side effects. Because I personally believe that, the best drug for a population would be the lowest and most mild amount of side effects. But as I mentioned, more than half of the drugs within the market have side effects, which is unfortunate. But as research continues, we hope to lower those. And do you have?
It was more of at the end of like researching the topic about obesity and the pharmaceutical.
Yeah.
Is it more like, are we, from what you're seeing like the data points, are you seeing that we're more pushing towards like using medication to treat obesity, or should we actually be taking a more different approach, like using the drugs to take off the weight and then promoting healthy lifestyles as the time occurs?
Yeah.
As the treatment trial occurs.
So I would say that would probably, now I don't necessarily know the exact data behind that, but I would say that would be the most effective way, because as we've seen and research has shown, if you are considered obese and you lose a relatively large amount of weight and you don't uptake that healthy lifestyle, you'll actually rebound further past that mark point of obesity, which will only worsen. So my understanding would have to be that, yes, use these pharmaceutical grade drugs in order to lower those obesity levels, and then we need to ensure that we're uptaking that healthy lifestyle, because there's probably 20 McDonald's in the area. That's, you drive down the road and we just see too many fast food and the declining food quality. So, thank you. Anyone else?
Thank you very much.
All right. Thank you, guys. Thank you.
All right, our next presenter is Isabella Presque from Mercyhurst University, talking about single factor B as a potential regulator of high salinity-induced conditional bacteria
Filamentation, sorry, the lighting's
That
bad
is-
for reading.
That is a mouthful anyways. Also, just like you say, I'm so sorry. PowerPoint hates me. So I'm gonna try and talk you through this. Thankfully, I don't have many words that you'll have to read, so you'll just have to listen to me, I guess. Anyways, hi, I'm Bella. I'm here with Mercyhurst University today, and like Jeanette said, I'm here talking about high salinity-induced filamentation in bacteria and sigma factor B as a potential regulator of that. But at the heart of the issue is the concept of stress. And I don't know about you guys, but sometimes I just really feel like this guy. Just everything's on fire and I'm so stressed out, but at the end of the day, you gotta just take what comes and adapt to it. And at the end of the day, we as humans aren't the only ones that have to do that. Bacteria do too. So, bacteria have and face a number of stress responses every single day in their environments, and they respond in a number of different ways. For most bacteria, they end up being survival strategies. These stress responses have to end up being survival strategies. A lot of them aren't, hey, I have an exam on Monday and I haven't studied at all. They're like, oh, this is like too high and I can't live. So, at the end of the day, the functions of every bacterial stress response comes down to restoring cellular homeostasis, increasing bacterial survivability, and making sure that these cells can face this stress again in the future in a better way. So, like I said, there's a number of different stress responses, and they're unique to every single bacterial species and the stress that they're facing. And at the end of the day, the stress responses come down to a modification in genetic expression. So, for example, I have two examples here. And again, I'm so sorry, it hates me so much. But, two examples here. So, number one, reactive oxygen species. So, this is going to be like your superoxide anions and like hydrogen peroxides of it. If the bacterial cells encounter a lot of these in the environment, they're going to increase, certain ones are going to increase SODA expression to help them deal with this, to help them manage the stress that they can survive and reproduce and go on to live independently. Same with heat shock. If the temperatures increase super quickly and a lot of bacteria will increase the growing expression to help them manage the stress. So the majority of genetic modifications that occur under stress occur here in stress. Specifically in bacteria, many occur through the changes between the interactions of RNA polymerase, our favorite friend, who is responsible for most of genetic expression in bacteria. It's responsible for translating all of the DNA into RNA so that the cell can do what it needs to do. So it's between this RNA polymerase, the catalytic core of it, and the sigma factors that come in and bind. Sigma factors are associable subunits in the cell, and they bind to RNA polymerase, and basically what they do is they direct it to certain promoter regions and genes. Basically, that's what our words say they act like the cellular GPS, and forward it up. So, there's two main categories of sigma factors. There's housekeeping sigma factors, and then there's alternative sigma factors. So, housekeeping sigma factors, these are your everyday sigma factors. They're going to help you do the basic cellular upkeep and metabolism, help the cells grow under normal conditions. Two most notable ones are sigma factor 70 in E. coli and sigma factor A in alternatively, the alternative sigma factors control transcription of specific genes. So they redirect the RNA polymerase to smaller promoter regions and other genes. Usually it's in response to stress. It's not all the time. These are like, I'm going to take over because we need to build connection. There's a couple of examples of alternative sigma factors. For example, sigma factor F, W, G. Sigma factor B is especially important in the and also, here, even though the alternative sigma factors might seem like they like they're smaller, they only deal with minor promoters, they will have a really big impact on the cell, not only because they control the stress responses, but also because they actually control a lot of genes and regulators. Like currently, the estimate for sigma factor B in regular members with the genes under control is like 255. So they're not as small as you would think. So I'm going to introduce you to one specific type of stress that I use in my research today, which is also it's a very common stress that a lot of cells have to face, which is salinity. So when any sort of cell, whether it's bacterial or plant, this figure is actually from a paper about plant cell stress, but it's so ubiquitous between the two for the most part that it's really illustrated to the yeah. One of the reasons so number one, so this is basically responding to the fact that which is more worried about the concentration of the solute in the ions and the effect that it has on the waters on the cell. So that's when you're going to see the cells blowing up, getting smaller, like that kind of thing that you learn about in like Bio 101, you know what I mean? There's also oxidative stress, which again, calling back to our reactive oxygen species friends like hydrogen peroxide. These can also be caused by high salt concentrations in the cell. And basically, the cells have to respond to this to restore homeostasis and get back to salt tolerance. And this is done through a number of genetic modifications. One specific salt tolerance stress-induced modification, too, is filamentation in bacteria, though the exact mechanism is still not exactly known. So, what is filamentation? Filamentation is a bacterial stress response. It's a type of abnormal cell growth where the cells continue to grow laterally but don't divide. In most cases, septal walls are not formed, no daughter cells are produced during filamentation. So what causes it in terms of like a molecular mechanism, like I said, that's still kind of unknown. Literature has implicated many genes in this process, mostly in the model species E. coli and Bacillus subtilis. And many are related to normal cell division. So here is my beautiful table of all the genes that I could find in the literature that have been implicated in one way or another in filamentation. I don't know about you, but that's stressing me out a little bit like that. So I took all that information, made a working mechanism / model for it. Yeah, that still doesn't look great. So I'm going to break it down a little bit for what we do know about the mechanism and what causes filamentation or where these genes could be implicated. So, in that, we're going to meet my friend. This is Bacillus subtilis. Like any bacteria, he wants to reproduce. But to do that, he has to undergo binary fission. So, he's going to copy his DNA. So, and then here is our mid cell. So that's exact middle. So at this point, there's going to be this gene, which when it gets just transcribed, is a membrane-bound protein that ends up recruiting other genes to form what's called the Z ring. Now, the Z ring is a site of cellular constriction during binary fission. So, some other genes, I don't know what happened to the like I said, powerful. Some other genes that go into the Z ring formation include FTSA, and which directly recruits FTSL to form the Z ring. And then as the Z ring constricts the cells to start forming two little baby cells, the FTSL protein helps to form the peptidoglycan cell wall so that the two cells can separate and become a daughter cell. And in literature recently, the gene MRAZ has been implicated in this process too, specifically in promoting the activity of FTSL. And if everything is right, two daughter cells are produced. But what happens if stress happens and this process is disrupted? Well, that's when you get filamentation. These daughter cells aren't produced, and instead, These really long, hair-like, weird cells are clones. Now, what exactly causes this filamentation? What genes specifically are involved or affected that trigger this filamentation is still widely unknown. The overall mechanism isn't known. How these genes interact isn't really quite understood. We have some basic information to understand it, but we're still in the beginning stages of putting this story together. Which brings me to my research. So in this study, we aim to identify key genes involved in stress-induced filamentation in a model species Bacillus subtilis, using a reverse genetics approach. And we also plan to use this information to try and propose a working overall mechanism for filamentation that other researchers can use to kind of test their genes and stuff so that we can start getting the full overall story. So, also to talk about reverse genetics, basically what we know is we have Bacillus subtilis. We know it filaments under stress, and specifically under high salinity. So, we know when it has all of its genes, it forms this filament phenotype. You knock out one of those genes, now it doesn't filament. That's what we're looking for. That was our goal in our first part, which is the genetic screening of our knockout mutants. And this is our kind of little roadmap. So first of all, we just looked at the knockout mutants. We had to identify them, confirm our results, quantify them. We also did a time course induction of filamentation and a Western blot. So first of all, we took 27 strains of Bacillus subtilis, the wild type 1A1 and 26 knockout mutants, and inoculated them into liquid culture, either tryptic soy broth or tryptic soy broth with 10% sodium chloride in it. Grew them at 37 degrees overnight in a shaking incubator. And then the next day we came in, we pipetted 20 microliters of culture on a clean little microscope slide, rubbed it all around so that it wasn't just all lumpy in one spot, and then heat fixed it and stained it with just crystal violet for 20 seconds, rinsed it off, visualized with a compound light. So these were our results. So these are all of the genes that we tested. So all of the mutants, those are the genes that they're missing. So they're all singular knockout mutants. As you can see, we had two results that were negative for filamentation. Up at the top, SIG-B, and down at the bottom, DAC-A. In this presentation, we're focusing on SIG-B specifically because we have not had the opportunity yet to really get into DAC-A and repeat and confirm that lack of filamentation. So that's kind of on the agenda for the future, but for right now, we're just going to focus on SIG-B. So after that, we repeated that screening process, so growing it overnight with 2 different cultures, heading liquid in the microscope, to confirm the filamentation deficiency, and we found that the mutants that lacked the gene SIG-B were continuously lacked the ability to filament. So over here, these are my beautiful microscope photos. So left side, that's all the wild type. Right side is SIG-B. And then in each of those, the control is on the left, experiment, so grown under high salinity, is on the right. As you can see here, you can see that the cells get a lot longer and they almost look like spaghetti. That's my favorite way to compare it. I think I was just hungry when I started using that analogy, but alas. And over here, they're a lot shorter, but I will admit, even blowing it up as much as I could on this slide, it's still kind of hard to see. So we're going to blow it up so that you can see it a little bit better. So, especially because over here with SIG-B, it can be kind of hard to see because some of these look like one continuous cell. But then here you can see with the filaments, they are continuous. You don't really see any like distinct little notch between them where they're separate cells. Well, here on SIG-B, where when it's smaller, you can see these ones, they look like they're one continuous cell, but they're actually separate, like distinct cells. So after this, we wanted to take these results and quantify them because let's be real, it's great to look at. Let's get some data in here. So we did the same inoculation, same procedure as before, and photographed them using the Modic PhotoPlex 3.0 microscope camera. They also have an in-house like measuring system and like calibration system. It was really cool. I got to play with it. Anyways, took those photos, did the measurements, and did all the data analysis and graphs me in Excel. So here are my results for all 3 trials that we analyzed. In purple is the wild type 1A1, in green is SIG-B, the SIG-B mutant. So as you can see between the control on the left, so the darker color, you can see, I'm also going to scoot ahead a little bit. So those are the average lengths, cell lengths in there, but I'm going to make it a little bit easier for you to see. So for 1A1, you can see that there's a significant like average cell length under the salt stress, versus in SIG-B, they remain pretty much the same. So I did the math, and the average percent change in cell length for the wild type is almost 400%, while in SIG-B, between the stress test and the normal conditions, like the control conditions, the average percent change was only 14%. And then, again, that's with the filaments, you can see visually, it just confirmed our visual results. So from this, we decided to do a time course to see if we could see when filamentation starts happening, to make sure too, maybe SIG-B causes filamentation early or something like that. So we took liquid cultures of both, did the whole procedure of the overnight stuff, and then to sterile test tubes, we took 3 milliliters of culture and 3 milliliters of the test, which is the TSB. In this case, we used 20% since we were adding 3 milliliters of culture of TSB that already had like some TSB in it, so it was diluting it. And then starting at T = 0, every 2 hours, we took samples we took a milliliter of culture to see for future protein analysis and RNA analysis, and then also we pipetted and did the whole staining procedure to visually see them. So here are our results for T = 0, 4, and 8, because as you can see, by T = 8, by the 8th hour, filamentation was only starting to occur even in our wild type. So if future directions is to repeat this process but have it be longer, because I think it takes a lot longer than we were expecting it to. But still here, you can kind of see that these cells on the bottom of the SIG-B are a lot smaller and haven't started filamenting, while the wild type definitely have. So after this, we took some of those samples that I said that we saved from T = 8 and did a Western blot analysis using standard protocol and the GRO-EL and SOD-A antibodies. These are all of our codes and treatments because I could not write all of this on pictures on our side. So basically what we did though is we used a T = 8 samples for control and 10%. We also had control in 12%. We have samples tested with 12%, mostly because there's another researcher in my lab who's doing research on the same mutants. Well, she's doing SIG-A, and she found something interesting that it can survive up to 12%. So we wanted to test it out just so a little bit of a curveball to bacteria. So as you can see here with the GRO-EL antibody, and then the SOD-A antibody. First of all, the T = 8, the time course antibodies did not work very well for mine, but I only used 0.5 milliliter of sample versus a full milliliter. But you can see GRO-EL activity goes completely away under stress, and SOD-A is actually inducted pretty heavily here under 12% high salinity. So basically, in conclusion, Bacillus subtilis mutants deficient in SIG-B not only visually demonstrate a lack of filamentation, but quantitatively also show a lack of filamentation, with a difference of 400% in the wild type and 14% in the SIG-B deficient mutants. Time course comparison demonstrates that the initiation of filamentation for these 2 strains do not occur at the same time. And in fact, 1A1, which again, SIG-B, we have not seen filamentation, so our working hypothesis going back was like maybe it happens early. Again, we would have to do another time course study to confirm this, but it does not seem like that unless it can speed run it super duper quick. But that's for future direction. So, and then the GROW-EL Western blot revealed distinct lack of GROW-EL expression in samples in high salinity, while SOPA revealed that there is an induction in the high salinity samples. So, what does this mean for the field as a whole? Why should we care? Understanding filamentation can not only help us understand that process, but also the process of cellular growth and division as a whole. And also, filamentation can play a really crucial role in a lot of pathogenic bacteria's life cycles. For example, uropathic E. coli cells that cause a lot of UTIs. They play a very vital role in not only their life cycle, but their pathogenicity cycle. So, future directions, again, I want to do a longer time course and also do qPCR and Western blot analysis with that. qPCR of the 8-hour time course. I also would like to investigate some of the regulon numbers of SIG-B to see if it's like further down the line too than expected. And then I would also like to do a genetic complementation test for SIG-B. So here are my references. And I would just like to say thank you to you all, and thank you to everybody in my wonderful lab group, because they're all awesome, and I couldn't have done this without them. And also, oh no, PowerPoint's messing up again. No, actually, thank you to you. Thank you so much for listening to me talk about this, because I really like to. So, any questions? One question?
Real quick. So I did a little bit of research on like BCA and effect of oxidative stress on H2O2. When you guys use NaCl, like is that all across the board causing all three forms of stress? It was only like specific to oxidative stress or when we did it, but I was just wondering.
As far as I know, it can cause all three kinds of stress because it's kind of like I said, with the osmotic stress and the ionic stress, they're so like interrelated because the ions are there. So the cell is going to react because those ions are there, but they're also going to react because that concentration of those ions is going to impact the concentration within the cell and outside the cell and impact how they have to regulate their water intake. I honestly have not looked into it much myself, which is something that I do need to do. But I would assume that the ROS species would also be affecting kind of like all at once. I would assume that it would be kind of like you're all at once getting affected by everything kind of thing. Thank you, Isabel.
All right, our next presentation is Ryan Zimmerman from LECOM, and looking at the comparison of UV light on colonial growth on cell phones. Ryan?
Perfect. First, I'd like to thank the RCSC for giving me this opportunity to present this forum to present my research. And like I was introduced, my name is Ryan Zimmerman, and I am a second-year medical student at LECOM, or Lake Erie College of Osteopathic Medicine. And my first question I'd like to ask you before I start presenting my research today is, what is one thing that all healthcare providers use on a daily basis? And that's either your doctors, your nurses, your PAs. That's your phones. Now, think about the last time you went to the hospital or doctor's office. How often did you see them on their phones while you're just walking around, or actually in the office with them right there? Like, this is a prevalent issue of how much they're on their phones. And the big thing is that we're going to actually take a look at it, is that antibiotic resistance is on the rise, and so is nosocomial infections. And if you don't know, nosocomial infections are hospital-acquired infections after a 48-hour time period. And I focus on Staph aureus bacteria during this time because focus on Staph aureus bacteria because it is a common prevalent bacteria that is seen in infections in skin and upper respiratory infections, and it's gram-positive. And I focus on E. coli because of the gram-negative bacteria that is commonly seen in UTIs, which it alternates between surgical wounds and UTIs for the most common nosocomial infections. For those of you that don't know, Staph aureus, as you'll see throughout my presentation, is that Staph aureus, as it does an infection, likes to grow in multiple massive colony groups because that is actually how it forms the infection together, while gram-negative bacteria doesn't need that many in the bloodstream to actually cause the infection. And you'll see that later on as I show pictures throughout the presentation. So the purpose of my research is to determine the effects of UV light on sanitizing Staph aureus and E. coli colonies throughout the phone cases, because like I mentioned, this is a massive issue that as future healthcare providers, we need to actually take a look at and further protect our patients. So to start my research, the methods is that I first overgrow bacteria overnight in broth, and then as I pull the broth, I have to realize, okay, how do I know how many colonies are in each bacteria sample? So I did a serial dilution to figure out the CFU colony counts. CFU colony counts are just colony forming units, and to do that was I pulled the broth. I would then check the OD to see if it was between 0.0 through 4 to 0.004 in the OD spectrometer, and then I would do a serial dilution, first doing the control, and then doing 10 to the 4th, 10 to the 5th, and 10 to the 6th. Now, up on top above is going to be your Staph aureus, and that's your control, through 10 to the 4th, 10 to the 5th, and 10 to the 6th. Same thing on the bottom, you'd have the control for E. coli, 10 to the 4th, 10 to the 5th, and 10 to the 6th. Now, you may be asking, okay, how many colonies were actually in throughout the presentation? And on average, it's about 10 to the 7th. There was a variety of ranges where it would be like 2.2 times 10 to the 7th or 5.5. But between both Staph aureus and E. coli, it was both, there were about 10 to the 7th number of, 10 to the 7th power number of colonies on my plate throughout each of the experimental procedure. Now, going into the actual experimental procedure after I did my CFU colony plates is that I would then first sanitize the phone in 3 steps. First, I would take the phone case and run it under lukewarm water with soap. Scrub it down for about a minute, 2 minutes. Then I would take an ethanol wipe and spray just basic ethanol sprayed onto a paper towel and wipe that down for about a minute, 2 minutes until it was air dried. And then I would then place it into the Pneumomed portable UV sterilizer for 3 minutes. Now, I wasn't sponsored or had any deal with Pneumomed portable UV sterilizer. It just is a basic UV box you can get at your local CVS and Walgreens. And I leave it in there for 3 minutes. And you may be asking, why would you do 3 minutes? Well, that's just what the box performs. So I'm not going to add any extra variation variables when we're going to be testing to see if the box is effective at its normal rate. After that, I would normally change my gloves, continuing keeping the sterilized environment, is that I'm putting 10 microliters of the bacteria onto the phone case. And I'm taking a basic sterile cotton swab and swabbing it down, starting in the middle, working my way up, and then working my way back down and letting it air dry. And that took about 15 to 30 seconds. Now, this is where you start to branch off between my control group and my experimental group. For my control group, for the no UV light, I would be done. I would take a sterilized sponge and I would swab the phones, and then I'd place it on the BBL Chrome Auger MRSA 2 for Staph aureus, and then the EMB or azinovin methylene blue auger. And you may ask me why you're doing different kind of bacteria. It's just because Staph aureus likes to grow on that one compared to the EMB likes for the E. coli. After that, I would then kind of swab it with the sterile sponge, swab it onto the plate, and then I would put it in the incubator for 37 degrees overnight. For the experimental trial with the UV light, continuing with the same steps going through it, sterilizing the phone, air drying it, I would then put it into the Pneumomed portable UV sterilizer for 3 minutes. And after that, change my gloves again, kind of keeping everything as sterilized as humanly possible, is that the phone case was kind of continuing the same process. Phones were swabbed with a sterile sponge, swabbed into place for Staph aureus, and then put on the BBL Chrome MRSA auger 2 for the E. coli, the EMB, the azinovin methylene blue auger. Once again, phones are incubated overnight, and the number of counts counted the next morning. And now this is a picture of the UV box. As you can tell, there's nothing really glamorous about it. It's just basically what you expect. The one interesting note is that right there is a UV light, and it's just a basic UV LED light. Now look at the results. Just by first glance, you can tell that no UV light over here, there's a lot of colonies compared to the UV light over here, to the UV light experimental group over here. On average, it was about 1,200 to 1,400 colonies on the no UV light that I had to individually count, compared to the 200 to 400 UV light colonies for the Staph aureus alone. And this is kind of going to what I said earlier, is that with the Staph aureus colonies with gram-positive bacteria, you see a lot more colonies forming because that's just their cell morphologies, that it's easier for them to grow in certain environments, and you need a lot more of them for actually causing an infection in a host. With the E. coli, and it's kind of hard to see because of the red, those are your E. coli colonies right there. Very small, very limited, but as you can tell, they're still kind of doing basic of the E. coli of like using their biofilm to form together, is that this is the no UV light and this is the UV light plate. For the no UV light plate, there's about, I would say about 100 colonies when I did the whole entire trial, compared to the UV light plate where there was 0. Now, actually breaking down my results is that there was complete 24 trials, but it was actually 12 trials with 2 sample sets in each size. And for the Staph aureus colony over here, there was an 80% removal rate between the no UV light to the UV light. Compared to the E. coli, which there was same thing. There is a slight caveat with E. coli that there were some outliers, but you had to, it was still statistically significant even without the outliers. So it came between 19 for UV light and 17 for no UV light. But before the outliers were removed, it was still statistically significant, and then they further just made it more statistically significant. And it was at 71% removal for the E. coli. My conclusions is that UV light sanitized the colonies of the Staph aureus and E. coli, which was clearly seen, is that the Staph aureus was more decontaminated at the 80% of the Staph aureus, while the E. coli was percent removed at 71%, and they're both statistically significant. And the results show that a simple UV light box can sanitize colonies of Staph aureus and E. coli. Now, you may be asking, what are the future directions? Well, the first thing is that this is a simple UV box that you can get at your local store. We need to test this if we're going to actually put this in hospitals with a more medically grade UV light box. And that is going to be that would be the first initial step. The second initial step is actually then implementing this into a real life situation on a hospital floor or in a doctor's office and testing, okay, are we still getting the same data that we are in the controlled environmental procedure? But also too, are the physicians, are the healthcare staff actually adhering to the policy too of actually putting their phone into the box? Because based on the data, we are seeing that it should work in a hospital, in a real life environment setting. But the problem is then we're adding the human factor. Are they then going to leave the phone in there for 3 minutes? Are they going to clean the box as much, like sterilize the box as much? And depending on how that goes, then I would like to actually put it into a full hospital, like a hospital multiple floors, to see, okay, if we're taking these extra steps to protect our patients, are we seeing a drop in nosocomial infections? Are we actually seeing that this is going to be worth to actually save patients' lives? Because at the end of the day, we're having a harder time treating the Staph aureus and E. coli because of this antibiotic resistance. And like I previously mentioned, UTIs are one of the most common infections you get in the hospital. Staph aureus is almost any infection that you get is caused by Staph aureus, and as MRSA is on the rise too. We may not have vancomycin to treat it any longer. We may be running out of treatment options there. So as healthcare providers, or as me, a future healthcare provider, I need to take the steps now to further protect my patient's care. And then my gratitude is I like just thank researcher LeCom for giving me the opportunity to research over the summer, the Research Collective for help funding my research, Dr. Cardy, Dr. Keller, and Dr. Waters for actually helping me with my process throughout this whole entire procedure.
You have time for questions?
I'm just curious, where did you source the phone from actual hospitals or was it your own?
So it actually worked out perfectly. I had to get a new phone, so I just used my old phone case that I would just use, and it was just your basic Otter box one. And that iPhone, since majority of people have some kind of like smartphone. That's what we kind of base it off of. And besides that, it also was it was an older phone case, so it was well loved too. So it actually like had more, it was more of that like well-worn usage that actually is going to implement more of like, is the bacteria actually going to stick to the phone surface? Mhm.
Does that UV light cost?
So I do not know the exact figure of the simple UV light box because Dr. Ferretti provided that during the pandemic for us to do research on. But I would, if I'm going to take an assumption here, and I could be wrong, probably the simple UV box is between anywhere from $20 to $40. The medical grade one is a little bit more of a harder sell because when I was sourcing images, this box right here costs anywhere from $2,500 to $3,100, which that may be more of a harder sell. But also, too, kind of going back into it is, it's not, from preliminary data right now, it shows that it's not the UV box that's going to be the issue. It's going to be the humans, because how many times are you able to actually convince a surgeon to show up earlier on the time, right when their surgery is? How many, are they going to be willing to spend that extra time to put their phone in the UV box for 3 minutes at a time? Are you going to be willing to see the actual support staff leave their phone in the UV box or put it in there for 3 minutes while they're on their break to use it again? It's going to come down to more of, is the hospital going to first implement the protocol, and are they then going to enforce the protocol? Because I could see them implementing the protocol and then, hey, we're doing our best job, and then not actually enforcing it later on.
Any other questions? Thank you, Madeline. Great, so we are having an early lunch break today, but please note that after lunch, we have presentations all the way up until after 4, so I would ask that you stick around for that. The boxed lunches, so if you do have to leave, please make sure you grab your boxed lunch before you go. It's across the hall right by the registration table in the cafe. And just to make you hungry, it is turkey bacon Swiss ranch wraps, or the veggie option is herb roasted vegetable wraps with balsamic, portobello mushrooms, red pepper hummus, etc. There's also drinks across the hall. So everybody meet back here at noon, please. Thank you.
Yo. Hello, welcome to the Regional Science Consortium 20th Annual Research Symposium. My name is Jen Salem. I'm the Regional Science Consortium Plant Lab and Greenhouse Manager, and I'll be your session chair for this halfway launch session. Our first presenters are Dr. Dinesh Nars and Ashley Pastori, and they are presenting the art of science, a multidisciplinary approach for all students.
All right. Well, thank you for all being here. Appreciate it. Thank you, everyone. We launched a really exciting program this year, so I'm excited to share this with you. And you know, even during the break, get some of your feedback as well. So as a scientist, and you're going to see this is where art and science meet, but as a scientist, if we look at the history, we can look all the way back to Einstein, and he said the greatest scientists are artists as well. And so I find it really interesting that as scientists, that art has been incorporated in the field of science for hundreds and hundreds of years. And you can see just from naturalists that had to make sketches of what they were looking at in the natural world to kind of depict these things that were accurately scaled. And a lot of the advancements in science have relied on creativity and visualizing things that couldn't necessarily be seen. So if you think of astronomy, how they are trying to visualize what is happening in space and adding color to that, and trying to interpret what is going on. Or if you think of at the microscopic level or at the cellular level, the different processes and the different things that happen have to have some visualization that maybe you can't see just by observing under a microscope or whatever, depending on where you were at in history and what type of equipment you had available to you. So both art and science require creativity, imagination, exploration, innovation, and visualization. And so I think it's interesting that we have all of these overlaps between these two disciplines. However, if you look at schooling, that K through 12 school, usually the students really get separated at some point. Like, oh, this group of students likes the language arts and, you know, the different art disciplines, and this group over here likes the different STEM disciplines, and they kind of get separated into different classes and doing different things. And what we want to see in this program, in this initiative that we're launching, is really keeping them together, bringing them back together, and meshing them to work together. So these students would be collaborative. So the art of science is embracing and encouraging the fusion of these two disciplines. And it's creating experiential learning where science and art are used cooperatively. And so even if we bring the students in, we're not separating them like come to the lab and do a science thing with us and then go to the art studio and do an art project with Ashley. We want to work together the entire time and show how these two things overlap throughout the whole process. So we also want to demonstrate teamwork. And you know, teamwork at all different ages is not always easy. But what we want to convey is that a diverse team is a robust team. So if you have a team of all the same type of scientists, you're all going to think the same way and have the same perspective. However, if you have a diversity of people on your team, an artist, a historian, a scientist, an engineer, you have all these different perspectives, and solving a problem or working on your project might get advanced a little bit further or in a different way. So this multidisciplinary programming is encouraging more student participation. So when we bring a group of students out, some students are kind of like, I'm not really a science person, this isn't my thing, or I'm not, you know, really into it. So we want to be inclusive of all students, saying that you might be interested in this area, art or science, and we're going to make it enjoyable and a good learning experience for everyone that attends. So what we want to do is also secretly instill characteristics of collaboration, communication, content, critical thinking, creative innovation, and confidence. So all these big C words, again, that is a good learning tool for these students
depending on if they're in the elementary, middle, or high school age. That's all really important learning skills that they'll develop. So in the fall of 2024, which is now, the RSC, in collaboration with Rounded Print Shop, has launched the Art of Science initiative for our K-12 education programming. Again, multidisciplinary and encouraging all students to participate. So if we go back in time, almost a year from now, I happened to get this email that said, hey, come to a workshop at Grounded Print Shop. And I was like, what the heck's Grounded Print Shop? I had never heard of it before. I just never noticed. So I asked my daughter, I'm like, you want to go? It's just a one-night workshop. It looks like it might be cool and kind of interesting. And that's where this collaboration started. I met Ashley, who is the owner and artist at Grounded Print Shop. And I was like, she's kind of an interesting person. And then we started talking, and she was saying how she makes paper. And then she said the word Phragmites, and I was like, you know what Phragmites is? I was like, you're an artist, and you know a word that is in my scientific field? That's awesome. So I felt like right there, we had the perfect connection. And if you don't know what Phragmites is, it is this big, tall, invasive plant that we have worked for over a decade trying to remove from the park. And so when we remove it, what do we do with it? When we remove all these invasive plants, what do we do with them? And so we formulated this Art of Science, which is a 3-part series. So the students come out and visit us 3 times in the fall semester. And their first visit, when they come out, Jen will take them out into the wetlands. She'll talk about wetland importance and function and all the work that we're doing there, and how invasive plant species have really compromised the integrity of these wetlands. And we get them to start to remove some of these invasive plants. And so these are some of our students here. They were taught how to identify mugwort, a type of invasive plant. They're bagging it, and typically we would just send it to the landfill. But now we can send it to Ashley at Grounded Print Shop. And so you can see Ashley's right here with all the students. I think having that integration of the artist being on site when we're doing science, as well as the scientists being on site and working with the students as we're doing the art, is what is a really good example in making this a very complete program for the students to understand. So once we bag it all up, we send it over to Ashley, and I will let her talk about the processing.
So paper making is a process. It is very involved, and we just made it more involved by actually processing the plants. So usually whenever I make paper, I actually at Grounded, we do a lot of recycled paper. So it's post-consumer, mixed office paper, cotton t-shirts, linen bed sheets, cotton bed sheets, stuff like that. I have an industrial shredder, and so we shred it all up, and then it goes into the beater. But this adds like 4 more steps into the process. So we're harvesting the plant, and then we are drying it. So that top, how do you use this thing? Drying plants up here. And then we take it to the chippers. And this, I mean, how many hours have we chipped plants? With the help of Haley and Sarah and Sean, and I mean, this is like a huge effort. How many hours do you really think?
Over like 20.
Probably like 20 hours of chipping, but that's all based on like how much we're pulling out. So it's kind of cool that we have that much to process. Then it gets put into some kind of, I just bought a cauldron. We were using pots, but now we will be using a cauldron to cook the fiber over a fire. That's cooked with soda ash to help break down the lignin, I believe, in the plant. So paper making is, you can pretty much make paper from anything that's high in cellulose. So I feel like we are learning which plants maybe work best. The Japanese knotweed is one that we tried, and the mugwort. There's just like so much mugwort, but both of those made really nice sheets of paper. I think that the mugwort is probably higher in cellulose, but I don't really, we should test this. So it gets cooked over the fire, and then we strain it out, and I rinse it, and then it goes into the beater, which this is the beater over here. So I have a 5-pound paper beater. I weigh out 5 pounds of dry plant or paper fiber, and then it goes into this machine and goes around in a loop for, you know, anywhere from 45 minutes to 5 hours. And that affects like the consistency of the paper, whether it becomes transparent or if you can see any bits of the plant left in the pulp. So day 2 of Art of Science, everybody, day 2, everybody came to Grounded, and we formed sheets of paper with the mugboards, and I think that we mostly just used mugboard. I don't remember if we had Japanese knotweed for this day. But we use a tool called a mold and decal. Has anybody made paper in here before? You guys have. Mold and decal is basically a frame. It's a 2-part frame, and what it does is sift the paper pulp or the paper plant fiber out from the water. Then we take it over to these sheets of Pellon, which are kind of like a interleaving or like a stiffener that you would put into like a collar or something like that. We use it over and over again, and it's what we use to stack up the paper. So you press it onto that. It's kind of hard to explain without like doing a demo. Then it goes into the hydraulic press. All the water gets pressed out, and the pulp stays where it is, which changes the paper from kind of being unstable to like more of a stable sheet. It makes the fibers like bind together, and then we're able to transfer them to the drying boxes where they dry for 18 to 24 hours depending on the humidity. Here's the process. It's everything I just described. So mold and decals up here. These are the vats. You can pigment paper pulp. So we experimented with, I know I can pigment recycled stuff, cotton, any of the stuff that I'd used previously, but I wanted to check and see if we'd be able to do it with the plants. So we did that and made it kind of a nice blue. This is unpigmented, and this is pigmented. These are students pressing the wet sheets onto Pellon. So this is the Pellon right here. This sheet. This is also Pellon. And then this is the hydraulic press, and this is the dryer. So that's the very long process. So at Grounded, we make lots of different kinds of things. I mean, it's paper. You can, what do you do with paper? People always ask me, what do you do with paper? And I'm like, what do you mean, what do you do with paper? It's like, well, we use for everything. But we specifically do like notebooks, and I don't think I have any, these are notebooks. Envelopes. I have like I have a die cutter, so 2 different size envelopes. These are some light boxes that I made with a fiber called abaca that comes from the Philippines, which becomes translucent when you beat it in the beater for longer periods of time. There's some examples of this in the hallway out there. This is a big decal box. Actually, this was when my grandma passed away a year ago, and we saved the flowers from her funeral, and we had her ashes, and my mom wanted to put it in paper, so these are her flowers from her funeral, and put her actual ashes in there. So that was, I think everybody felt like that was a very, it helped them kind of process a little closure. I don't really know if I can fully describe, but I think that it was a beneficial thing. And the paper ended up drying really beautifully, and it will be passed out to everybody. So yeah, I sell paper on Etsy and in person, and I guess we're kind of deciding how to deal with the paper that we make from the invasive plants here. But it basically can act like a normal sheet, and so I think that part of the importance of adding art into pretty much everything that we do has to do with being able to have space to think creatively and wonder about anything, because we can find new solutions and different pathways to achieve anything that we want to make happen. And so while I think that there is a very concrete thing that is sellable through this collaboration, I think that maybe the even more important thing is that people are able to see a process from start to finish, and ask questions and kind of wonder, well, what happens if we do this, or we change this, or, which is essentially what I'm doing with my business. I'm just wondering about things and then going down that path and trying to explore, which is why it's so cool to work with scientists, because I don't know the answers to a lot of things. And next up, we are going to be processing pigments with the students. And like I said, this is still very much an exploration for all of us. But this process is called, sorry, I just felt like I was talking for a long time. I don't know how, okay, good. So it's called lake pigmenting, it's a lake process. So what we're doing is taking plants, various parts of the plant. It could be roots, it could be bark, berries, and extracting a dye from the plant, and then adding a mordant. So in natural dyeing, you have to add a mordant or you have to like wash your fabric in a mordant, which allows pigment to bind to the fabric. And in this process, we are adding a mordant essentially to the water that has the color, the pigment in it, and the pigment binds to the mordant, and then we are able to separate the pigment from the water, making it an insoluble material that we can then use for making ink or watercolors or any kind of paint, which this is all very exciting to me for obvious reasons. So this is some silky dogwood berries, which were very blue. They were like a very bright blue. And then when they were processed, they actually turned into, I think that this is the one that it was just like a lime green. So I'm learning a lot right now, and with the help of everybody here, we're learning a lot. And I think that it'll be really interesting to kind of potentially use this color as a way of mapping an area, because even over time, you could take the berries from one plant in one spring, and then the next spring, the color will be different because it is very affected by the water, like whether it rains, what the soil pHs, all of this stuff. There's so many variables, which really can create like a beautiful range of color to work with, and also tell us something about the environment. Here's some more. I keep talking about the things before I show them. So this is, well, let's see. So this is this is after we've extracted the dye and we've added the aluminum sulfate to the dye stuff, essentially the dye liquid. And we let it sit, so the pigment will settle on the bottom and the supernatant will rise to the top. And then we strain it. So this is like the first strain. It gets cleaned maybe 2 or 3 times, and then you let it dry. This is what it looks like whenever it's drying. And I was so fascinated by this. I have just left them sitting around because they're so cool. We've taken these plants and boiled them and broken them down, and then they reorient themselves into these patterns that look like a leaf or something like a plant. Like, how is it doing that? It's very fascinating to me. So it dries, and then I scrape it all up into a pile like this and grind it up in a mortar and pestle, or I think I'm gonna get a coffee grinder. Then it should be able to be mixed with various oils, depending on the application, to be used for painting, or I'd like to see if I could add this to paper pulp, pigmented paper pulp, because that's what we're doing. So come visit. Grounded's a cool place. We have paper making classes, relief classes, bookbinding, open studios. We have maker markets, and so Grounded is a collection of artists and business owners. There are people that rent in the building. There's 2 buildings, so we're kind of creating this community of people that all have different skills and bring different things to the table. There's a yoga studio at Grounded now, Studio Sage. Check it out. They're really cool. The space is really beautiful. And we actually have a paper making workshop this Saturday which I need people to sign up for. There's a paper maker from Kent State. She's a grad student. She's teaching that on Saturday. We've got some bookbinding classes coming up with a master bookbinder in January. And also, Open Studios are now on Mondays, so I think that the hours are like 5 to 8, and it's a space where you can come and see what Grounded's about, bring stuff to work on, or learn something new. We do have intro to carving, relief block, usually on those days. Yeah, I think that's it. Did I miss some stuff?
No, I think that was great. Does anybody have any questions?
We do have time for a few questions.
And tried using grapes, there's plenty of grapes around Northwest Pennsylvania.
Yeah, I actually have grapes growing along my fence, and I keep forgetting to go grab them to try it. There's also kind of a shelf life to this stuff, so I like had all these plants and all these different things, and I'm like, oh yeah, we're gonna make pigment, but it takes a while for the pigment to drain it's like through a coffee filter. And so I need to come up with some solutions to maybe store things while they're draining or drain things quicker because they'll mold, and I end up having to dump things.
We also have a lot of winemakers in the area.
Yeah.
And one of their processes is to allow this during fermentation, all that sediment that you're looking for comes to the bottom, and they throw it away. And
That's a good idea. Man, that is a good idea. And I actually just watched a video recently about this girl in Oregon who she was talking about the plants that she was trying, and one of them was a native grape out there, or a grape that is grown out there, and she said that it failed over and over again. And I didn't get to finish watching that video, but I was like, I wonder why. Because sumac is one that we have tried a number of times. So you would think that in the berries, so when we extract the dye from the sumac berries, it's red, it's like fuchsia. But then when we add the aluminum sulfate, actually it doesn't change when we add the aluminum sulfate, right? Do you remember this? And when we add the soda ash, it turns to like a very dark blue, like gray blue. And we've tried it like a number of times. So, I mean, and I definitely think that it has to do with the pH. Like, so we want to achieve a, what's it called? When it's neutral. Yes, a neutral, like solution. And with those berries, they're really acidic. So I don't know if we're just not, I know I'm like using these words like, I'm not sure.
See all the science that is, like the chemistry that really goes into this, and even the temperature plays a role. And so part of what we were thinking is as we try different recipes to get to the end product, that we can actually run the color through a sample and a spectrophotometer and get a reading on it. And so then we can start kind of classifying them as different shades or different colors depending on their reading, which would be the scientific way to measure it because we all see color differently in different shades.
Also the pH, I mean, like I learned how to use a pH reader thing. Yeah, with the solution and whatever. But that was really helpful because we could see right then and there, like, we need to raise the pH or lower it or whatever. Yeah. This is all stuff that I think that
So much chemistry.
Yeah, but
so much art. So.
Yeah, yeah. But it's like very hands-on, so I feel like I don't know, it's like relatable to lots of people, or you know, people that maybe don't. Jacob was over there doing math, and I was like, "That's great. I don't know what you're doing at all, but yeah."
And when we bring students there, like the pictures we were showing where they were pulling the paper, that was a group from the community college, and it was also some high school students that are there for dual enrollment. So we had college student age as well as high school age, and they literally lit up. This was like a great experience for them where they were like, how do we do more of this? How do we, you know, they were really inspired. So I felt like this, you know, initiative that we're launching has just a lot of potential, and this is just paper making and dye extracting are just like the first two steps, and so I think it's really going to grow.
Yeah.
Thank you.
Thank you.
Oh, thank you for doing that. So our next speaker will be our own Jacob Court, and he is presenting on experimental place-based learning on Lake Erie Regional Science Consortium Ship to Shore Program 2024 to 2025. All right, come on up, Jacob. I know.
All right. Thank you, Jen. Yeah, this is a tongue twister. Experiential place-based learning on Lake Erie Regional Science Consortium Ship and Shore program for this academic year. So first I'd like to say thank you to Jeanette for taking me on here. I'm probably the newest member of the RSC. Just yesterday I was really officially hired. So I've been working for a few weeks, and this whole project, sort of the broad scope of it was put together by Jeanette, and Haley Dahl here has been doing all the fine tuning. So I'm really presenting the work of other people, just so you know. So the Ship and Shore program is a new program to the RSC this academic year. Here's a flyer that Haley put together for advertising for this program. There's Haley here teaching at the Bayfront Maritime Center. The goals of this program is to provide an opportunity for students to learn about science and the environment using active learning approaches, hands-on learning, to get their hands dirty or wet in the field. And it's a place-based approach, meaning we're going to learn about the place that we live in and we grew up in our own community. Another goal is to participate in high-level laboratory techniques. So we'll talk more about that, but the students are participating in lab techniques that have been developed by or approved by the EPA, their standard operating procedures. So this is like real science lab process. In the end, so the end goal is for students to create a stewardship action project. They are going to create their own project to solve some sort of environmental issue on their school's campus, with the goal of this being learner-driven. So they're going to do their own research, put together their own project, and present on it at the end of the academic year. Another goal is to promote the understanding and appreciation of the environment in which we live. So we want them to gain an appreciation of Lake Erie, of Presque, of aquatic ecosystems, and how we, as people living here, might impact those ecosystems and environments. Another goal is for us to sort of abide by this framework developed by NOAA, this educational framework called MIWI, Meaningful Watershed Educational Experience. You can get trained in the MIWI educational framework. I'm only about an hour into many hours of training on this. So if you have any questions about that, we may direct it to Haley. More overview of this program. It's implemented mainly by us, the RSC, but we are getting help from, again, the Bayfront Maritime Center team. I'll talk more about what they've done for us, and the flagship Niagara League. The grant for this program, all the funding, costs a decent amount of money for all these things. It's the Pennsylvania Department of Environmental Protection providing the funding. And our target audience are students ages for grades K through 12, their teachers, from our member schools. Okay, overview of the program. I'm going to go through the different parts, what we're doing. It's divided into 3 major parts, but there's also a pre-exercise, pre-field trip exercise that the students did. There's a manual I'll pass around. But part 1 included going on the Levy G. Howard ship, sampling water from the bay, from Presque Bay, and learning about maritime science. Part 2, the students are going to sample water again. They haven't done this part yet, but they're going to sample water the lakeshore or in wetlands on Presque Isle. They're going to analyze their samples from part 1 and part 2 in the lab. And they're going to also learn to identify phytoplankton, and they should and macroinvertebrates. And then lastly, again, the implementation of the stewardship action plan. Okay, so these are the pre-field trip exercises that the students are required to do. So Haley put together this webpage with the help of some of the other team members here at the RSC. So this is the web page. There's the link there. That's kind of what it looks like. The students can log into this page and complete different exercises, learn about the environment, learn about water quality, macroinvertebrates, the different type of testing and analysis that we might do. It also introduces the students to the Ship and Shore program in general. The RSC team talks about watersheds of Pennsylvania, especially the Lake Erie Basin, the one that we're in. Testing methods, and then there's this video. I have to hand it to Haley and the team members here. They put together this really excellent video. It's 20 minutes long, and it covers all of these different types of water quality analysis. Talks about Lake Erie. I'm just going to play a little sample. Hopefully this will work.
Fellow student scientists, and welcome to the Ship and
Can you
Shore
hear that at all?
program for the 2024 to 2025 academic year. Throughout this program, we will be assessing water quality on Lake Erie, and you will be working with staff from the Regional Science Consortium.
Okay, so that's just kind of an introduction to the program, but there's also part of this video shows different types of water quality analysis.
So these are the macroinvertebrates that we sampled from Marina West on 7/29/2024. So what we do is collect the macroinvertebrates with a net and put the vegetation in a jar with ethanol, and then like Ken is doing.
Might be hard to hear that, but that's some of our interns from last summer showing how to use that macroinvertebrate sampling.
Yep, they were taking macros.
Very cool. Yeah, the great thing about these videos is they can watch and learn about the entire program before they do the hands-on part of it, and all the background. Additionally, there on the website is a pre-assessment Google survey. So this is how we're going to assess the learning outcomes of this program. Twice we found out. Hold on. I'm just going to advance. I just picked out a couple of questions from the survey just to highlight some of the things that we asked the students. Well, one of them is just their grade that they're in. So 38 students responded. That's not everybody. I think we're still working on getting a couple more. But most of the students in 8th grade, middle school age. This question here, on a scale of 1 to 10, 10 being the most important, 1 being the least, how important is a healthy watershed to the local community? The students pick what they feel is the importance. Most students, I think it's encouraging, they picked 10. But maybe by the end of this project, we can shift that curve more towards the 10. Another question, can you identify a local environmental issue either in a specific ecosystem or in your community? So 63% said no, they couldn't to that question. And then if they did say yes, they answered sort of what was a specific environmental issue. Most students answered some form of pollution, climate change, erosion. Interesting to see that. One student answered tornadoes. So hopefully by the end we'll have more responses to that question. And then lastly, what would you like to learn about in your community in terms of environmental issues? And so they could type in whatever thing they wanted to learn about. And this is a good way for us to respond to their wants and needs. We can answer these questions along the way in this program. So some interesting questions I picked out that the students asked are how some of the toxic algal blooms form on Presque Isle, which I think we did kind of address in part 1 a bit, but we'll have more work to do on that in part 2 in a couple of weeks. What are some endangered species in this area? What is water quality like in the lake, and how does climate change affect us, or how does it affect us since we're not in the ocean or not on a shoreline or a desert? So how does it affect people in Erie? I like that question. Another great resource that Haley put together here is the program manual. We're just going to pass them around if you want to look at them. So the students actually get something physical to hold on to write in, take notes in. It's got all sorts of information, again, about water quality, the environment, lab analysis. It talks about, again, watersheds, freshwater ecosystems, and it's got a step-by-step instructions for all of the different types of lab analysis that they're going to be doing for this project. And there's data sheets. Again, all this stuff's pretty high level, so I find it really amazing we have these students doing this. For it. All right, so the main part 1 of the program was a voyage on the Letty G. Howard tall ship on Presque Isle Bay. So we took the students on the ship. They got to learn how to sail, the basics of sailing. They learned some of the basic boat parts and directions. You know, I got them confused a bunch of times. Couldn't even tell where the front of the boat was. Again, this was operated by the flagship Niagara League. They were very, very good, very friendly. The students had an opportunity to hoist the sails and sail across the bay. So here's some pictures there, the students hoisting the sails. I think I have a little video. I guess this is, a lot of people in Erie do this, but I'm not from here, so I've never done this before.
You're gonna see it going up?
Right, also on the tall ship, on the bay, we had the students collect their own water samples. And using EPA standard operating procedures, we used these plankton tow nets to capture water, hopefully get some of the plankton that they can identify later in the lab. We also used the Kemmerer tube. You've ever seen one of these? It's this long plastic tube that you drop in the water. It's open on both ends. Once you get to a depth that you want, the two ends, you drop a weight, the two ends snap shut, and it captures a column of water. Pull that up, take the sample back to the lab, and at a later time you can analyze the water. And then additionally, we had them test for water clarity or turbidity with a Secchi disc. You drop this disc down in the water in a line, allow it to sink in the water and, until you can no longer see those white and black wedges. The deeper the Secchi disc goes, and you can still see it, the clearer the water. We also took other analyses too, I don't have pictures of. Water temperature, weather conditions, all the sorts of stuff that are important for proper analysis. That same day, the students, we took them back to the Bayfront Maritime Center, where they learned the basics of ship science. We had instructors showing them, or demonstrating buoyancy, water displacement, density, and ship design. So you can actually float a ship on water, and they learned the science behind it. Once they were done with that lesson, they got to build their own model ships out of just various types of paper, cardboard, plates, clay, all sorts of materials, with the goal of trying to float it on a wave tank. That's the wave tank there. So this board moves back and forth, creates these waves, and their ship either sinks or floats. They designed it right. Additionally, they learned the basics of knot time, the knot time lesson. All right, so that was part one. That's already been completed. We already got some of the water samples. For part two, we're going to do more water sampling, this time offshore instead of in the bay on the water. So we're going to collect more water samples, biological samples, including macroinvertebrates, using what these are D-nets, is that right? These things are kick nets. So they're going to catch macroinvertebrates, so we take them back to the lab, they're going to identify them. They're going to learn to use dichotomous keys, so they can identify dragonfly larvae or mayfly. Additionally, they're going to learn about the importance of indicator species. So if you have mayfly larvae present, hopefully that means the water is fairly clean. It indicates something about the environment. I did not have pictures of our students doing it because they haven't done it yet. So these are actually college students doing these sampling. They're going to take their samples back to the lab and do different types of water quality analysis, which includes testing for bacteria, some coliform bacteria, like E. coli and Enterococcus. One of the methods, the one that they're going to actually do, right, is the vacuum filtration process. You take your water samples, basically filter them under suction. So it filters quickly. The bacteria stays stuck to the filter paper. Then you take that paper and you put it on some auger plates, incubate it, and see what grows. Then they count the colony forming units and they can tell something about the quality of the water. Another test that we're not going to have them do, we'll talk about, is the IDEX system, which is much fancier, expensive. They're not going to actually do that, but you get to see it. Some other sampling that they'll do is, or testing, is testing for HABs, harmful algal blooms. So mainly in Lake Erie, it's kind of a misnomer. It's really bacteria, cyanobacteria, that causes most of our HABs. But HABs float. So one of the tests you can do is the float test. Just shake up a sample of water, see if the algae floats. If it does, it's probably a HAB. That's my understanding. They're also going to learn to identify different algae under the microscope. They're going to do other water quality analysis, dissolved oxygen, nitrates, different nutrients. And then finally, for part 3, which we're still in the process of fully designing and implementing, the students, again, they're going to design their own environmental action project. So this could include, actually they're going to look at their school's campus, look at an environmental issue that they'd like to solve or address. Could be like stormwater runoff, recycling, invasive species, They'll come up with a plan to help solve that issue or raise awareness for the issue, implement the plan, and then report back to us on how that project goes. Conclusions. The progress of this program will be reported in January at some point. And students are going to present the results of their Stewardship Action Project at the end of the academic year. We'll take another Google survey to see what the students have learned. And they'll also write a reflection of their experience and how they think it may have changed their view of the environment. And I'd like to thank again Haley for a lot of the work she put into this, and Jeanette, the whole RSC team, it's been great, and member schools and these other people. Any questions or anything? Yes?
Did you say there were 120 students?
How many total were there?
Off the top of my head, I think it was around 100.
It was around 100?
Yeah.
About 100 students?
And how were they selected? Did they self-select or were they?
They have to be a member school for the RSC. And then from there, I'm actually not sure.
So we have different member school districts, which I'll talk about in my presentation a little bit more. But basically, I have a whole network of different teachers, and then it's a lot of administration, gifted coordinators in different school districts. And so I send out all the flyers with all the information. And then registration is first come, first serve. In the past, sometimes we had a hard time filling that up, but in my presentation, you'll see we did not have that issue this year. So that's kind of how we recruit our students for those programs.
Thank you, Jacob. And we have Haley Dahl presenting next. It's another RSC team member. And she's going to present the 4 seasons of STEAM. Let me get your presentation.
Thank you, Jen. Sure. All right, well, I'm going to explain myself briefly, but Jacob did a thorough job doing that already. Thank you so much for all that attention. That was great. But also thank you to Jacob. He's been a great help this year, and we're really grateful to have him because, as you guys are going to see here, our education program has expanded a lot this year. So the help is very necessary, and we're super grateful for that. So I'm going to be presenting on the 4 seasons of STEAM, which is actually the 2nd annual year. That's the grant that I got hired off of last year. I started last September with the RSC as the education manager. Last year was the 4 seasons of STEM though, so I'm going to kind of describe what that looked like last year and then how that's kind of changing this year. And then I'm going to tell you guys all about our education program too. So we've got a lot going on this year. All right, so last year we were branded as the 4 seasons of STEM. So we had the science, technology, engineering, and math, which is what a lot of our programming and education stuff is focused around. As you guys know, we have introduced art this year as well. We also only did 3 seasons last year because of some funding things that were going on, and I also started a little bit late, so the programming was a little bit reduced. So I have these kind of categorized here. The first part, students joined us at the Haggerty Family Events Aquatic Center, and we kind of showed them some scuba techniques. We were joined by Divers World, and students actually got to put on scuba gear, learn about the different parts of scuba equipment, and go underwater, test out those breathing apparatuses. We also used our PVC shipwreck skeleton, and students looked at trilateration techniques of measuring a shipwreck. And then they also did some inventory using snorkeling techniques. Then we moved on into the spring, and we used Presque as our resource for this, took students out there. And as most of you probably know, there's a lot of erosion that's going on and present, so it was a good resource to use where students could see that happening firsthand. So we did some erosion and coastal resilience activities with them right on Presque over by Beach 7. We talked a little bit about tombolos and then some of the mitigation structures and prevention for erosion that occurs on Presque. And then over the summer, we had the same students join us for our 4 Seasons of STEM summer camp, which we had help with Gannon from. And they did a lot of different stuff. It was a really busy 2 days. They did some invasive plant removal on Presque and some native plant planting. They also did fishing, and then they did some water quality sampling on the Lety as well as processing in the labs, and then they got tours of the lagoons. Something I wanted to mention is we have been trying to introduce in all of our programs the use of using a scientific field notebook, and it's always cool to see how students interpret that. So we always give them an overview. Here's what this can be used for, your observations, and what they choose to write in there is always really different. But I like this picture because we were talking about fishing, and we were also talking about some bacteria at the same time using vacuum filtration. So I really liked the drawings of in this field notebook. So that being said, this year we are reintroducing and kind of revamping as the 4 Seasons of STEAM. But something unique this year is that for all 4 parts, we have 4 unique locations. And the 4 Seasons of STEAM was kind of introduced to represent the different seasonal changes apparent in Erie, because that's something that with our local place-based learning opportunities, students get to experience all 4 seasons right here in Erie, which is pretty unique compared to other regional areas. Something else that I wanted to mention, like I said, the different locations. I think that there's a lot of value in providing these different locations because it provides a different experience to students. So I'll get into the first part, which students already attended, but we went to Grounded Print Shop, and there's a flex space up here. And I think that there's a lot more creative learning that can happen there. There's a lot of artwork and a lot of open space, and there's a lot more independent learning and connections that students can make there. And then obviously we're using the Haggerty Family Events Aquatic Center, and students get to be under the water, so very unique environment there as well. And then this year we're actually using a new location over in Harbor Creek. It's near Shades Beach, and it's the Glenoto Center, which is across from the monastery of the Benedictine Sisters. And here's just some pictures of when we toured there. There's a really big open space to gather the students in, but there's a lot of spaces within the property that they can be outside in nature, which I think is really valuable for the experiences that we provide, because students are able to make those connections to nature while being outside. And then obviously we've got our beautiful resource right here, Presque as well, and all of our research labs. So this year, like I said, we already did our first part, so there are 4 parts of this. Part 1 is the art and nature exploration, which happened at Grounded Print Shop, and then we are going to head into the winter season, where we will do some scuba stuff, which will be a little bit revamped. In the spring, we're adding something completely new, some agriculture and technology. And then the last part will be a summer camp, which I'll get into, which will be very different from what it was last year. So we had a really good time over at Grounded Print Shop, and I'm gonna kind of explain how that looked. So kind of blending that art and science again, but I don't want you to get confused, because we have Art of Science 1, Art of Science 2, and then we also have our 4 Seasons of STEAM. But we're kind of trying to incorporate art everywhere we can, because like they were saying, art has always been apparent in science. So we're focusing on the blend. So the students came out, and we actually had Tyler Brooks from the Natural History Museum come out and show us some scientific techniques of preserving plants and making herbarium sheets. So after that training he provided for us, then we kind of turned it into more of a student program where they can learn the step-by-step process of that. So as you can see here, those are some prints from the actual Tom Ridge Natural History Museum. And this is a buttonbush species that we use during our training. And then the students got to press different plants that we kind of thought were part of this fall season. So we were looking at different leaves that from Presque. So we were doing cottonwood and sassafras and red maple, and there was a few other ones in there. But that was a step-by-step process, and they learned how to do that in the presses. And something that's kind of cool to mention, this is an actual, once they're all pressed and strapped down, this is to dry the plants. But we ran out of space in there, and Ashley's way of drying her paper at Grounded Print Shop is actually pretty much identical to how we do it for plant pressing. So once again, the blend of art and science, and it was very convenient that it was just there for us and readily available. So one of the things these students did while they were here was, like I said, with these field notebooks, they created their own field notebooks, as you can see in this bottom left corner, and the covers of those are actually from that mugwort paper. So everything's coming full circle over here. And something that was cool about that too, as you can see in this photo, students got to use an old-fashioned typewriter to type out their labels for their field notebook. The last part of this first session of the program was stamp carving. So every herbarium sheet gets its own stamp for the museum that it's being stored in. So we had one created for this program, but then students got to take their own pieces of rubber and carve their own stamps. So these were some freehand ones, but I thought they were really cool because the student was trying to reflect the different categories of trees, which tied into the whole program as well. So that was part one, already happened. Part two, so the shipwrecks, scuba, and snorkel. I thought that this year we would do that again just because last year all the students really enjoyed it and there's been a lot of outreach for that specific program, so we're going to utilize that resource again. But we're going to revamp it a little bit. So with the upcoming designation of the Pennsylvania waters as a NOAA marine protected area, we kind of were brainstorming, well, how can we get the students involved in this as well? So we have our past team, which there will be a session tomorrow. You guys should attend that if you're around. But we're going to be working with some people from there. We actually have some members of the RSC team who are on the past team. Me, Sean, and Sarah, and Jeanette are all part of that team. So we have those firsthand resource for the students to use. But they're basically going to come out to the pool and we are going to show them actual archaeology techniques, which we use to document shipwrecks in Lake Erie. And then once again, they're going to be using different methods, not just the trilateration, we also might do some different sampling methods with them as well, and then do some snorkeling techniques. And then part three, going into our spring, this is a brand new program that we're introducing for this, a brand new part for this program. Last year we did the erosion, this year we're doing some technology and agriculture. So we thought, what better way right here, we were talking about some of the vineyards. There's a lot of different agriculture that happens. So for example, there's some small urban farms that work to get out produce to the local communities. So Wildfield Farms is someone that we're working with. We're looking to work with Welch's. We just kind of want to demonstrate how that's apparent in this area and also how innovations in technology can be apparent on small scale farms and local farms all the way to your big corporate farms as well. So this is very much a work in progress. But one thing we're excited about is, I don't know if you've ever heard of the forward education models, but they have a microbit processing chip that is compatible with their devices, and we are looking to purchase a classroom set of these because they are all based around climate action. So students will get to use coding and different technology techniques to kind of brainstorm solutions in agricultural practices. And something else that's really nice about these kits, they come with all the lessons already prepared, and you can tweak those however you want, but they're already mapped to all the standards that we use, including the PA Steel standards, which if you do any type of education, you know how important those are. And then the last part we're very excited about is our summer camp. Whereas last year we did all the programming and Gannon did all of the overnight accommodations, this year Jacob and I are putting on our backpacks and sleeping in tents so that these kids can have a really good camping experience, I hope. But we're actually going to be staying out by the lighthouse and doing a bunch of different things, including some night hikes, some cast iron cooking, and then obviously we'll put together our scientific programming as well, like sampling and fish seining and everything on Presque. It will be a very naturalist approach to learning, and we're excited to kind of see how the students will be receptive to that. All right, so that was all the 4 seasons of STEAM for this year, which we're really excited about. Off the top of my head, I think we have 80 students registered for the full program. They have to attend the first 3 sessions, to come to the summer camp because the summer camp is limited registration. But for the other programs, we're opening that up to our entire member school district network. So anybody who wants to attend those first 3 can. They just have to come to all 3 to come to the last one. But obviously, we're advertising it as a consecutive program. Now I'm going to get into the rest of our educational programming for this year, kind of explain how that works and how it's changed as well. So I'm sure anyone who's ever been aware of the RSC education knows about the Forensic Science Escape Room. This year is actually going to be our 8th annual year, which is exciting. This one is probably one of our most sought out events. We actually dress up and we do some filming for that. And we put together an entire storyline that then we use some graduate students who are in forensics departments, and they come up with the labs to supplement our storyline. So at the end, they can actually solve the murder, and it's really fun. We all get to dress up. But yeah, so we always schedule 3 days, and this year, I sent out the registration at the beginning of the year, and it filled up within the first 2 days. So we've actually added an additional 2 days. So now we've got 5 total, and there's over 300 students registered just for this event for this year. And we are at maximum capacity, obviously, but there are still people inquiring about attending, so there might even be some more dates on here in the future. But yeah, this is really fun. We, like I said, come up with the storyline. This is a scene that Mercyhurst's applied forensic science graduate students did last year. And we just kind of really like to show students that the forensic science is applicable in real life, and it's not just something that you see on like CSI and different crime shows. But this one is a really fun one. So I won't go into this too much because Jacob did a great job explaining everything, but we also have our Ship and Shore program, which we're really excited about. We have done this in the past, but this year it's a little bit revamped, and like I said, with the NOAA designated marine protected area, there's a lot more opportunities where we can get the students involved in the outreach for that as well. Another really sought out event that we do, this is our 9th annual year, is the Problem Solving Hackathon. So students will come in and they will be presented to by different researchers, and we choose a topic every year, and experts will come in and talk about that, and they'll take notes, and then they'll go through different benchmarks for the hackathon in a hackathon approach. So last year the topic was robotics, and then the students had to research an environmental issue and come up with a prototype using robotics to solve that issue. So we have cardboard and all recycled materials that they can use, and they'll actually design a prototype using that and present it to a panel of judges, and then awards are presented at the end. So here's just some examples from what students created in the last years, students with their awards. And in similar fashion to forensic, this is already at maximum registration, and we did have to add 2 extra days as well. So we're really excited about that. Something that we did last year and we're continuing this year is our fishing program. So the RSC actually got their training and serve as fishing skills instructors. And so we learned a little bit about teaching students how to fish. And obviously we have a great resource here. So we offer a few different topics, and they can either be, we'll set a schedule for them and students can get registered for those, or our member schools can reach out and schedule their own dates to come out to do this. So just a few topics that we cover with those are the adaptations of aquatic species, fish species of the Great Lakes, and basics of knot tying and gear setup. We also, during our fishing skills instructor training, got a lot of materials and resources from the PA Fish and Boat Commission, which has been very useful for us. We are also setting up a club at General McLean, so we are sending them all the materials, providing teacher trainings, and then they will have those resources available to do their own fishing club there. So yeah, here's just some pictures of students having fun fishing. This was, they were coming up with their own fish species based on different adaptations that we assigned them, and they would tell us how their fish was best suited for their environment. And then over here, we actually have the students collect some macroinvertebrate samples with that vegetation, and we kind of talk about the food bugs and why macroinvertebrates are important in a fish's environment. I'm not going to go over this in detail either, because obviously we had a whole presentation on it, but Aside from the 4 seasons of STEM, we also have our Art of Science program, and we had students already come out for the first part of this field trip. Once again, we were only scheduled these first 3 days. We had to add on another 3 in addition, just because everything's at such a high volume this year, which is really great for us. And the Art of Science 2, where we'll be looking at extracting dyes and pigments from plant material. And we actually are all fully registered for that as well, so we're probably gonna have to add some more dates. And that is all I have. We have a lot more education going on, but in this time frame, I don't think I could fit it. I have a poster that has way too many words on it, so if you're here later, you can ask me more questions about that. We're always looking to recruit new members, but for this audience specifically, if you have any ideas for our programs, or if you want to be a collaborator or come out and see what we do, we're always looking for that connection as well. That's all I have. Yes, Amber?
Just curious about your fishing program, if you incorporate any, like bait ideas for students who are talking about the importance of bait, because that is one way that aquatic invasive species are introduced. So that might be a great venue of collaboration between the AC grant and RSC, if you're not already doing that.
No, that's a really good idea. I mean, we do the basics of like night crawlers versus like bloodworms, but we also have kind of introduced using artificial bait as well. But yeah, that's a really good idea, and that'd be a good collaboration. Any other questions? Well, you guys know where to find me.
And we will be taking a very brief break until 1:15 today, and then we will resume our presentations. So thank you.
All right. Most of you saw this in the last session, but I'm Haley. I'm the Education and Outreach Manager for the RSC. And we're going to be into our third session of the day. So the first presentation is from Femi Aloy, and it's the evaluation of seed oils of economic importance.
Okay, my name is Ben Ray. I have my PhD in chemistry from the University of Pittsburgh, from the University of Aberdeen. This article I have here is just a presentation of this was an example of the work I did with some of my students at my other university before I moved here. Sorry.
It's the side arrow button.
Yeah, it's like I can't use that one very well, so I just want to use my one.
Okay.
Instead of turning. Yeah. So, quickly I want to take you through some of the highlight of that particular publication. And like I put here, if you want to know more about what I do, you scan this, you can see more about what I do here. Now, in the article that I put here, we have 3 seeds that the students decided to work on. And the reason why the students decided to work on those seeds is because every time they come into school, they see those seeds every time. And they were wondering, what can we use these seeds to do? And they approached me that, can we do anything with this seed? And then I told them, go research about it and let me know the name of the seed, what other people have done about the seed. And they said they not work on the seed in our area, but they work on some of the seed in other parts of the world. Then I said, okay, the seed might be the same everywhere, but sometimes, depending on the area where you are, the composition of the seed might be different from each other. Let's try it. So the students decided to look at the yield of the oil they can get from those seeds and try to look at what we can use the seed oil to do. Then they characterize the seed and see what is inside the seed. So those are things they did. So I just put the highlight of their observation here quickly. And from there they discovered that the fatty acid composition of those seed oil were different from each other. And then they tried to look at different fatty acids that were inside those seed oil to see what they can use them for. Then they discovered that one of the seed oil was active, as broad spectrum against different different microbial organisms, pathogens. Okay, like I put here, if you've been to, even here in Erie, you've noticed that there are a lot of plants, a lot of walkway where people walk, and you keep imagining, apart from we just going through the train, what else can we do with those leaves, trees, and other things that we have there? So one of our plans, or what we are doing recently, is to go inside the bush, get some mushroom, try to extract some essential ingredients, some active ingredients inside those mushrooms, some plants, get their leaves, try to see what are the things that we can have there. After we extract it, what we are intending to do with those extracts is to see, for instance, Do they have medicinal property? Do they have antiseptic property? Do they have any things we can use them to do? So that's what we have been doing lately. Now, like I've said, in the past we've characterized seed oil. We are still doing that here, and we are extending from just seed to other type of plants, and things like that. And to talk about seed oil, we all know what oil means, and we all know the importance of oil. And now that the world is trying to see the possibility of not relying on natural oil, there is this diversion of people producing oil and trying to use them for maybe biofuel, biodiesel, and things like that. But the problem with using oil for biodiesel is that there's competition with food. Then the oil will be expensive, blah, 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 people that are not rich might not be able to buy it. So, and that is why we are looking for alternative sources where we can extract those oil that are not edible. So we want to look at oil that are not edible that we can use for different industrial purposes. So now, like I put it here, seed oil can be used for different applications. Many of us knows that we use seed oil to cook. You have your canola oil and different oil that you have used to cook before. It has some nutritional value. And in our cosmetics, we use it, there's a lot of oil there. And for industrial purpose, we use it for biodiesel, we use it for lubricant. And from seed oil, we have some pharmaceutical application that we can use it for. Some oil, we can use them as insecticide. And we all know that pesticide, insecticide can cause a lot of hazard, mostly the artificial one. But the natural one that we get from seed oil are always better. We use it for surface coating, and that is why some of these our desks are shining. And we use them for candles, soap making, and things like that. So it has a lot of application. But the question is, which of the application can we use them for? To know the application we can use them for, we must know their properties, know their chemical consistency. But like I've said earlier, if we just use any oil, both one that are edible and the one that is not edible, the problem is there will be competition, food competition. So now, the first thing the student needs to do is to identify the seed or the plant. Then the next thing is it edible or is it not edible? Then the next thing is, what is the ethics? The area, will people be willing for that plant to be extracted? Because some places, they don't want you to use their plant for any industrial purpose. So after the students have identified that, when they are sure that they can actually work with the seed, then they need to know the name of the seed. They work with plant scientists to identify the name of the seed. Then we do extraction of the oil. We have different methods that we can use to extract oil from seed oil or from plants. I will talk about that after this. After we have extracted the oil, to characterize it, to know the chemical constituents, sometimes we do bleaching, and then we can send it to GCMS, GCFI, GCFID, and things like that. Then we can do the physicochemical analysis. These are different physicochemical analysis that the students normally do. Then we can do the antimicrobial activity to see if it has effect against some pathogens. I listed some pathogens here. Then we tested it for biodiesel to see can we use the oil for biodiesel. Here I listed some of the methods that we normally use in chemistry to extract oil. You have what we call cold pressing. If you think that the active ingredient or what you need might evaporate if you use it, then we use cold pressing, we just press it to get the oil out. Then if you think the cold pressing is not enough, that there are certain things that you see within your plant, you want everything out, we use hot pressing. That will give you more yield than cold pressing. If you want to go on larger scale, sometimes we use expeller, we use enzymatic extraction, just put some enzyme to help you to do the extraction. We use supercritical fluid extraction. Most times we just use CO2 here to do the extraction. Steam distillation. Currently one of my students is using this approach because she's interested in extracting a particular oil, which we call volatile oil or essential oil. And the reason why she's using this is because if she apply it, the heat might damage the essential oil. So she decided to just use water, and when she boils the water, the steam of the water will interact with the plant. Then the vapor will take up the essential oil. Then she can recover the essential oil after condensation. Maceration. This is a common one in third world country, where all what you just do is to soak the plant. Then after some time, you can separate it. Then use some other method of separating funnel to separate the extract. Then we have what we call solvent extraction. The common solvent that we normally use in lab most times is N-azine. We look at the polarity of the material we want to extract. If it's polar, we use polar solvent. If it is non-polar, we use non-polar solvent, because polar dissolves polar and non-polar is very good with non-polar. After we have gotten it out from here, so this particular one I put here is what we call sourceless extraction. So when you are heating the solvent, the vapor of the solvent will go up. There won't be a way for it to escape because there will be condenser put on top. It will condense it back to this thimble, and then it will keep washing this for several times, and it will be coming back. You continue the cycle until you are sure the extraction is complete. Now, for the students that work on this particular project, what she did is to pick the 3 plants, 3 different seeds she wants to work with. Then she blended it, and after that she used sourceless extraction. I just described sourceless extraction. Then she got the oil. After that, she tried to remove some aqueous material in the oil, and she tried to convert it to biodiesel through esterification, and then she got a biodiesel, then she characterized the biodiesel. So, to convert it to biodiesel, what she do, this is the reaction. The oil she got is what we call triglyceride. She reacts it with alcohol, and we get biodiesel and glycerol. After that, the next thing is just to separate glycerol from biodiesel. So this is schematic diagram of what we did for the esterification. The first thing is your methanol and your catalyst. Then you add the oil to it. After that, you separate it. Once you have your products, you separate it, and you have your biodiesel and glycerin. These are the 2 products that I said we're going to have here, biodiesel and glycerin. The next thing she did after she has done the biodiesel determination, conversion of the oil to biodiesel, the next thing is to see if the oil has any antimicrobial activity. So she selects some microorganisms, and here she used bacteria, she used virus, fungi, and vis-a-vis like that. What she was interested in doing was to see when she put a particular pathogen in a particular sheet and see if there will be a growth. If the oil will be able to inhibit the growth of those pathogens, then she will measure the length of the growth at a particular time. So quickly, let's look at the result she have for the oil. We discovered that the oil, the seed, has different yield. The question I asked them after they got this is that, is the yield enough for commercialization? 1. 2, is the yield close to what has been reported in other places? We discovered that the yield that they have, once the yield is greater than 40%, I think is of commercial value. Then, is it within the range that people have reported in the past? Yes, it falls within the range which people have reported in the past. We put the range in the paper that I will put the QR code to that paper at the end of the presentation so that we can look at it if you want. Now, looking at the physicochemical property of the oil. Different seed oil gave us different color. So the color of the oils were not different, were not the same. We are not expecting them to be the same. They are not the same material, but they came from different material. And if you have been to the store, when you see different oil, you discover that they are slightly different from each other, depending on if you are buying canola oil or corn oil. They are not exactly the same because they are not from the same source. Now we look at the pH. If it's too acidic, you don't want to eat an acid, of course. And so we want to know the pH of the oil. So we look at the pH, then we look at the specific gravity of the oil. The specific gravity of all the oil is the same. Why are we looking at this? It's because we want to know what can we use it for? What industrial application can we use it for? Then we have shown us the yield before. We look at the acid value. The acid value will tell us if the oil will be able to stay long or it will degrade quickly. We look at peroxide value, saponification value, free fatty acid, and we just try to compare them with the standard and be sure it's within the standard of oil, one that is edible, or it's within the standard of oil that are not edible. It's within the standard of oil that if we try to preserve them, they're going to stay for a longer time. Then we look at the moisture content. You can see the moisture content is very low, telling us that we don't have water inside those oil that we extracted. Now, we send the oil for characterization. We want to know the amount of fatty acid in the oil. The reason why we want to know the fatty acid composition of the oil basically is because we need to know what we can use the oil for. One, we need to know if the oil will be very reactive or the oil will be stable. We want to know if the oil can transit quickly or the oil, when you use them. I don't know, let me ask this question before I continue. When we are trying to cook, have we seen oil, bringing out foam before? If you are trying to cook, have you bought any oil that is bringing out foam? So those are things we want to be sure of. Is it anytime we are just trying to cook, then it's going to be bringing out foam? Or is it oil that will last long when you are using them? So when we look at them, we discover that the fatty acid composition of each of the oils are different from each other. And that is why when we are buying oil, even the normal regular oil in the store, it is always essential for us to look at the composition of those oils. So here, some important things that we discover. Let's look at this cysteine heptadecanoic acid. That's C17-1. Here, this TC is the only one that has that chemical in it. And when we try to use these three oils for antimicrobial activity, we discover that these two behave the same way, while this one behave different from these two. Then we concluded that the reason why this is behaving differently from this is because of the presence of this particular compound, which is the major composition of this oil. Then we try to look at other things, like what is the percentage of unsaturated fatty acid in each of them? What is the What is the percentage of unsaturated fatty acid in each of them? Then we just look at them. The reason why we are looking at them, like oil can be saturated, oil can be unsaturated. If the amount of unsaturation in the oil is high, the oil will be very reactive, which is not that very good. Because it means if you put it there, it can become solid. So this is how we did the antimicrobial test, putting them on this plate after we have incubated the plate and try to measure the growth with ruler to see if the microorganism is increasing or it's not increasing. Okay. And these are the results we obtained from the antimicrobial test. We have some control. We have two control. We have tetracycline. We have water as our control. And the reason why we are using those control is to see this is known like a medication, that people normally use to treat a lot of broad spectrum microorganisms, and this is just normal water. Then what we are trying to do is to compare the effect of our oil with this and with the distilled water. After we did that, we also tried to do what we call certain number, biofuel, potential, and higher heating value. How many of us know what we call octane rating? Every time we go to buy gas, we see something they call 87, 97, you buy different one. In diesel, we use cetane number instead of octane rating. So we try to calculate cetane number from our oil. And what we discovered is that the oil has different cetane number, which is very good, and because it's more than 100. We look at biofuel potential, and we can see they have high heating value and high biofuel potential, telling us that we can use the oil that the students extracted for biodiesel, and we can use them for different applications. Because of my time, I will not. These are just the property of the biodiesel that the student produced, and I stop here. So from what the student found, they discovered that one of the oil can be used, for as an antimicrobial agent, and some of the other oil can be used for biodiesel. So that's what the student found. And what we are doing in our lab currently is to look at different plants and see if we can use them for different industrial or domestic value. Thank you.
All right, we have time for one quick question if anybody has one, and then there will be time after this session as well if anyone has a question. All right, thank you.
Sorry, I like my stuff.
I did. Right, our next presenter is Holden Cooper, and he's going to be presenting on When Serpents Shine, Exploring the Biofluorescent World of Snakes.
Hi everyone, thanks for being here. So today I'm going to be presenting on the research I've done documenting biofluorescence in snakes. So before I get too far into it, I should probably address what biofluorescence really is. So biofluorescence is what occurs when an organism absorbs light and then re-emits this at a lower energy frequency. And I have an example of this with chameleons. So here's a chameleon just kind of in normal light conditions, and then there's a photo of it when being exposed to UV light. So what happens is it absorbs UV light, which is a higher energy wavelength, and then re-emits this through the bones, which you can see here, that's actually showed through the skin at the color blue. So blue being a lower energy wavelength. And it's important to note this is different from bioluminescence. In bioluminescence, an animal is able to produce its own source of light. So in this example, I have a deep sea jellyfish, and this jellyfish is able to produce this bright red coloration when it's being attacked by a predator. The reason it does this is to draw in other predators that may be larger than the one attacking it in this burglar alarm display. Since it lives in the deep sea, there's not much light down there, so this bright red is going to attract a lot of different things. Overall, a lot of what we know about biofluorescence is in more marine systems. And we have a lot of really good examples of what its ecological function is. And in this one, I have some photos of a cat shark. Cat sharks, which are found kind of deeper in the water, tend to hang out in environments where blue light can really penetrate the water pretty well, and it tends to be the predominant wavelength of light in that environment. However, these fish do not actually see the color blue, but instead are able to pick up on the color green. This coloration of this biofluorescence allows them to see other conspecifics and more effectively just communicate with one another. In birds, we have an example of more terrestrial fluorescence and its ecological function, which overall isn't as well documented, but in this example with parakeets, we just have a biofluorescent parakeet here. And what happened in this study is they were experimentally had their biofluorescence reduced by putting sunscreen on them, which is kind of what you see here. And when this happens, they did a mate choice trial, and they found that females actually preferred the males that did not have their biofluorescence kind of interfered with, kind of proving that biofluorescence in parakeets is used in some form of mate choice. So for what I wanted to look at, I wanted to focus on snakes because at the time there weren't many studies and still aren't very many studies focused on biofluorescence in snakes. And of these studies, many of these don't include spectral data. So the reason I'm really hammering the point of spectral data is because spectral data is going to be a numerical value associated with a color. So spectra is just a measure of differences in peaks and distances between them. So over here with these lower numbers that are going to be higher energy wavelengths, so we have UV, then violet, and go through blue, but we have numerical data that we can actually compare to other studies. So more studies should really be focused on this. And as I mentioned, there are very few studies really focused on snakes overall. There's only one actual manuscript that's focused on snake biofluorescence, while the remaining studies are more so just natural history notes that are more so documenting its presence. For what I wanted to do, I wanted to determine if snakes biofluoresce, and if so, what variation would there be between species, looking at both shed skins and live animals. So for shed skins, I focused a bit more broadly and looked more at family differences. So I looked at boas, pythons, Boidae and Pythonidae, Viperidae, which is going to be our rattlesnakes, copperheads, and cottonmouths, Colubridae, so that'll be rat snakes, garter snakes, and a lot of other ones that are pretty common in Pennsylvania, and then Elapidae, which are coral snakes and Australian brown snakes and moths. For the live animals, I focused more specifically on that family of Colubridae and looked at species that can be found in Erie County. And this family is broken up into 3 subfamilies, so Colubridae with our eastern milk snake, gray rat snake, and smooth green snake. I focused on 3 species of garter snake, the northern water snake, queen snakes, and red belly snakes, as well as brown snakes. For Dipsadinae, there's only one representative that can be found in Pennsylvania, which is the northern ringneck snake. To document biofluorescence, what I would first do is take a photo of a snake under white light conditions, so normal light conditions. Then I would take a photo of different body regions. I would do 3 dorsal regions and then 3 ventral regions, and I would expose them to a different source light. So for live snakes, I would only use UV, blue, and green light because those were the lights available to me at the time. And I would shine this light on the animal and have a filter placed in front of the camera lens. This would ensure that any of that light that I was exposing them to isn't getting through, and I can solely focus on what's being biofluoresced. For both shed skins and live snakes, we did the same thing, where for collecting spectra, but the only difference is we're using a spectrometer. And this is where we again get those numerical values. And once again, shed skins did not, or live snakes did not use violet or cyan light. So overall, did they biofluoresce? And overwhelmingly, the answer was yes. What we see is a lot of biofluorescence in between scales, particularly on the underside and near their vents. So the vent is their neurogenital opening, and this is particularly fluorescent in live snakes after they've musked, which is a defensive behavior they engage with. And it's pretty prominent to UV light and violet light and blue light. So for those kind of patterns of biofluorescence, we see 2 main ones in the 2 different subfamilies, and I have white light photos of different snakes in those different respective subfamilies. So here in Colubridae, we have a milk snake, then a rat snake, then smooth green snake, and over here we have a ribbon snake, short-head garter, queen snake, and then another short-head garter snake's vent. So the main thing that we see is that under UV light, and both blue and UV, is that the ventral surface is the most fluorescent. But in juveniles of Colubridae, you see these areas here are going to be more fluorescent than the kind of outer lip scales, and more so the chin shields is what those are referred to. And it's more pronounced under blue light, where you can really see those differences. But once they get older, you kind of see biofluorescence more similar to what you see on those lip scales. For the family of Natracinae, it's a very different story, where the fluorescence is in between all scales throughout. And you see this in all the different species, but to varying intensity depending on their life history. And we'll talk a little bit more about that in a second. For shed skins, we kind of have the same trend where, again, dorsally they don't show much fluorescence, but in between scales, especially near the lips, they're going to have a bit more. So this is UV light, and this over here we have a ventral surface of a snake under cyan light. And then once again, once you get further down the body, there's a bit less fluorescence. So this is just kind of like the mid body of a snake shed skin. And then here we have it exposed to violet light, and this is its vent. So when I quantified the spectral data for shed skins, I was able to look at the intensity and kind of compare those between the different families, since for this I would be keeping them at set different distances from the light and the spectrometer probe. And what I found was that under UV light, colubrid snakes would be the most intensely fluorescent, and whereas on ventrally it would be viperidae, so vipers, that were the most fluorescent. For live snakes, I'm not really able to do that same kind of analysis, but I am able to see how much variation there is with color. And what we see overall is that there isn't much variation between species or between individuals of species. So overall, why should we care about any of this biofluorescence? Well, largely we don't know if there is any ecological function, especially in reptiles. And some potential functions could be in mate choice. So for eastern garter snakes, they use a lot of visual cues during mating that tend to be less documented, since we mostly focus on more pheromonal cues that snakes pick up on during mating. That tends to be what science focuses on. But there is some kind of indication that these snakes will actually use different, more visual cues. And one kind of clear sign of that is that females, so in those famous mate balls that a lot of garter snakes kind of exhibit, the females right before they emerge and get bombarded by males will shed their skin. This fresh set of skin is going to biofluoresce more than more older skin that is about to undergo shedding. And I see this a lot in a couple of different species where their biofluorescent cues are kind of muddied when they're in this more, it's called in blue, where they're in this darker shade. Additionally, biofluorescence could enhance intra and interspecific communication. So in this central photo, I have an example of how rattlesnakes will defend their territories, where two males will raise up their ventral surfaces, exposing them to each other before they kind of interlock and try and shove the other one down. Since these snakes had really prominent biofluorescence on their ventral surface, having this enhanced biofluorescence here could be some signal of health, or honest signal of health that's showing that a potential rival might be in better condition than the snake fighting it, and just kind of give them more reason to back down. Another example for more interspecific communication can be seen here with this coral snake. So the Sonoran coral snake is known to engage in cloacal popping, where they raise up their tail and expose their cloacal linings and making a popping noise. Having biofluorescence here, and since I saw biofluorescence being more notable on snakes that musk, this having more enhanced biofluorescence could indicate to a potential predator that they smell terrible, to stay away, and kind of give them a visual cue as well as auditory and other cues. We kind of see more similarity with this too in some of the species that we looked at. So other snakes in this kind of same genus that the milk snake is in, so California king snakes, are known to expose the linings of their cloaca when they are musking, and this is having more fluorescence here as they're kind of showing that to a predator could potentially serve to further ward them off. Another reason they could be using biofluorescence is to enhance aposematism. A lot of predators of red belly snakes, shown here, include bullfrogs, and frogs overall, they tend to, in amphibians, have more green shifted vision and are less likely to see the reds that are on the underside of the belly. So a common display of these snakes is to show off that red belly. But if there's a frog that doesn't pick up on that as well, having biofluorescence that shows more green when they're absorbing more blue light that's in the environment could help them better ward off a predator. And there is some pretty good potential for that, since the time that both the frogs and the snakes are going to be more active is closer to dusk and dawn. And at dusk and dawn, we tend to see more blue ambient wavelengths of light. So having more blue light in the environment is going to give them more significance for that. Another reason we should really pay more attention to biofluorescence is because it could help us better protect species. So I mentioned earlier that life history really impacts how different species biofluoresce. And we see that within even subfamily level, where here I have, this is an example of a ribbon snake where it's really intensely fluorescent to blue light on its underside. But when you look at an eastern garter snake on its underside, it's not nearly as intense as this species. And this is because they kind of have different life histories. These snakes tend to spend a lot of time up in shrubs near more aquatic environments, whereas eastern garter snakes tend to be more generalist and spend a lot of time both in aquatic environments and terrestrial or more woodland habitats. So having this kind of difference in life history is going to better show us how these snakes are really using the light in their environment, and if we have things like light pollution affecting that, it could make them appear more visible to a predator. And then lastly, disease detection. So one disease that's really plaguing a lot of different snakes is snake fungal disease, which is a fungus that impacts their skin, causing lesions. And one thing that I noticed, especially while looking at UV light, is that it can kind of highlight those different regions. So here we have a milk snake where you can kind of see its lesions, but not super well. The photo isn't the best for it, but you can really enhance where those lesions are. They really show up under that UV, with the same being true for this gray rat snake. And these are my acknowledgements. Does anyone have any questions?
Any questions? We've got some time for them, so anyone has any?
I don't imagine that shed skin bioluminesces once it's separated from the snake.
What was that?
Shed skin, will that bioluminesce?
Yes, that was a big part of what I looked at.
Oh,
So
sorry,
there's an example of that here and here. So this is cyan light and green light. So it still retains that, which is pretty interesting. Yeah.
Is there a time frame that it retains it for? Like, would it start to degrade over like? I mean,
So-
I assume the shed also degrades over time. Does
Yeah,
it?
when we get some, basically, she's asking.
Yeah.
So for shed skins, I tended to just focus on the more recently collected ones, if possible. But overall, I didn't really notice too much difference between those older ones and the more new preserved ones. Yeah.
I'm going to miss this, but how do we know that snakes can see this type of biofluorescence? Like just
Yeah.
because they're doing it
Right.
doesn't mean that other snakes can actually see it.
Yeah, so snakes are able to see actually a little bit better than we can, where they are able to see UV wavelengths of light all the way through red wavelengths of light. So they're able to see all those different wavelengths of light. And in the case that they're using it for communication, it could really just be like this certain wavelength of light is hitting them, but then it's not getting reflected back, so then they absorb it and reflect a different color back. But also maybe it's not for that, and it's more so just for between a predator and prey relationship. Yeah.
So you mentioned like communication between the rivals and fluorescence being a bigger fitness. Do you know how like fluorescence actually like might be an indicator of fitness and what it's actually indicating?
Yeah, so if there's more intense fluorescence, and this is more just hypothetical, it's not an actual, there's no science backing this up yet because this is pretty novel, but having that more intense fluorescence could be indicating that they're able to get more. So like a lot of other vertebrates, when they have more carotenoids, it's kind of a signal that they're doing better, they're more healthy. It could be something similar to that. Yeah.
So what would be for you like the next step in this research that would fascinate you, like the questions that this raises that you would want to explore?
So I would be really interested in doing something similar to what was done with that parakeet study, where I'd conduct a more behavioral trial and kind of do a similar thing where I'm putting on sunscreen and seeing how different snakes will react to just different individuals and how that impacts mate choice.
Any other questions? All right, thank you, Holden. All right, and for our last, actually, I don't know if there's supposed to be another presenter after this. I'm not sure if they ever showed up or not, but this next one is going to be characterizing the gut microbiome of the river otter from Libby Babcock and Lorenzo Tavanche. All right, there's a clicker right here. The center is the laser if you guys want to use this.
Thank you.
Yep.
Yeah, so today we're going to be talking to you guys about our work characterizing the gut microbiome of the river otter in our local waterways. So to begin, this is just the timeline of our presentation. We're going to start with an introduction describing the river otters in Pennsylvania and just gut microbiomes in general. Then we're going to move on to our methodology, how we chose our latrine locations, our actual scat collection methods, and the general microbiome analysis after we get our samples. And then we're going to move into our expected results and finish off with our conclusions and our expectations for our continuing research.
So river otters are a semi-aquatic mammal native here to Pennsylvania. They are very social. Most pictures that you look up on the internet, they are in groups from 3 to 6. And they are very nocturnal, so they hunt during the night, except for the winter, where they will hunt during the day to avoid the freezing temperatures. Because they are social, they use many forms of communications. One form that they use is scent marking. So they will scent mark in latrines, which are a 20 by 30 meter area near a waterway. Well, they will excrete scat and anal jellies. There are high activity periods at these latrines during the fall and spring. They are active in the fall for territory and then in the spring for mating. And they use scent glands near the base of their tail to excrete hormones. And this is just a video of what a scat scent looks like. And we were actually able to get that from one of our sites that we studied. So what do river otters in Pennsylvania look like, and like what's their history? Historically, they were found in all major rivers and watersheds in PA. However, in the late 1800s and early 1900s, due to unregulated trapping, water pollution, and habitat destruction, their numbers were severely depleted. And there was only like a group of 6 to 10 individuals left in the Pocono region. And using the otters from the Pocono region and from across the country, the Pennsylvania Game Commission teamed up with Penn State University, to reintroduce 150 otters over the course of 20 years. So they started that in 1983 and actually finished that in 2004. You can see they reintroduced them to most of the major rivers and PA, and the Pennsylvania Game Commission came out in their river otter management plan in 2021 that the river otters are considered established and stable in Pennsylvania, and because of this, there's now a regulated trapping season. And currently, river otters are detected in all historical locations and are continuing to spread with minimal population density.
So to contextualize our research, it's important to actually know what a gut microbiome is. So a gut microbiome is described as a community of microorganisms, primarily bacteria, archaea, microbial eukaryotes, and different viruses in the gastrointestinal tract. And the gut microbiome has a significant effect on an individual's digestion, metabolism, and growth, and is also associated with different changes in neurological development and behavior, like mate selection, which influences obviously mating behavior. The gut microbiome is primarily affected by an individual's diet, stress, and exposure to pollutants through their environment, and that can create an altered microbial community composition, which is called dysbiosis. And that can affect an individual's homeostasis, make them more susceptible to infectious and autoimmune diseases, malnutrition, cardiometabolic disorders, and also maybe cause behavioral and cognitive impairment. And as you can see in this image,
the alteration of the microbial community composition in those individuals has a significant effect on all of those different factors. So to describe some examples of microbiomes in different mammals from previous studies, first I wanted to focus on Tasmanian devils. And in that study, it was described that the individual microbiome composition was heavily influenced by their carnivorous and scavenger lifestyle. So humans and mice actually exhibited similar patterns of the microbiome composition in these Tasmanian devils, which was associated with a high efficiency and energy harvest from their diet, making them more susceptible to developing obesity. So for wild Tasmanian devils, that's really important because they have to be able to really harvest a lot of the energy from the food that they're consuming. It was found that they consume up to 40% of their biomass in one day, and then they can go 2 to 3 days without eating. So that microbiome makeup was heavily associated with their need to extract as much energy as possible from their diet, which just provided an example of the diet and eating habits influencing the gut microbiome in those individuals, and indicating that the lifestyle of the individuals alters the microbiome composition just as much as the microbiome composition influences an individual's lifestyle. And the study actually indicated that carnivorous species are a lot more susceptible to microbiome changes, with factors including, like I mentioned before, their diet, habitat, stress, medical treatment, or different social conditions. And those alterations could produce physiological changes in those animals which influenced their individual health. Moving on more specifically and more relevant to our study, there was another study on Eurasian otters and snub-nosed monkeys, which demonstrated that animals that rely on one food source and a more strict diet have a less diverse gut microbiome. Since otters are more omnivorous than monkeys, the study pointed to a higher relative microbial diversity in those individuals compared to the snub-nosed monkeys. There was also a difference in carnivory compared to herbivory, with differences in the microbiome composition related to compounds in plants and bacteria that are needed to detoxify those compounds within an individual's gut. The microbiomes of otters were also heavily linked to the composition of the soil microbial communities that they were living in. It was described that animals are able to acquire different microorganisms through soil microbe exposure. Different studies have shown that mice exposed to soil microbes actually had a higher relative gut microbial diversity compared to mice that were not exposed to those soil microbes, demonstrating that the environment affected the gut microbial diversity within those individuals. Since monkeys are not living within such close contact in the soil compared to the Eurasian otters, that demonstrated another factor that influenced the gut microbial composition within the monkeys compared to the otters.
So why is this study important? River otters are a top predator in their ecosystems, and there are few published studies on the gut microbial community on river otters. River otters are very opportunistic hunters, and their prey varies by season. So during the summer, they mostly eat crayfish and frogs, while during the winter, they will eat fish and salamanders. River otters are also often a captive species in zoo and aquariums, which can implicate differences between captive and wild river otter microbial communities, which can also help inform better implications for wildlife rehabilitation. And then there's also the difference between beneficial and pathogenic bacteria, which depending on which bacterium we find in our samples can indicate the health of the otter population in the waterways, since they are semi-aquatic and spend a lot of time in the water. So what will we do with our results? This study will start a process of measuring the health of the river otter populations in Pennsylvania, more specifically the French Creek watershed, which is where this study took place. And it can help inform conservation management strategies that can be used to implement to improve the health of the river otter populations and the ecosystem. So the first step of this study was to locate river otter latrines in the French Creek watershed. So we searched previously known habitat areas. We used information from the Pennsylvania Game Commission, previous research done by Dr. Pierce, and wildlife enthusiasts who let us use their property for latrine searching. So in June, we were able to locate active latrines in State Game Lands 270 in the Erie National Wildlife Refuge. An active latrine is one where there is fresh scat and is used quite frequently. We did locate a non-active latrine in State Game Lands 214, which is just to the right of Linesville, or left of Linesville, sorry. However, because it's non-active, there was no fresh scat, and we were strictly using fresh scat for our study. In July is when we started our scat collection, and we searched for more latrines in State Game Lands 213, which is otherwise known as the Geneva Swamp or Geneva Marsh, depending on what you call it. However, there was no signs of active scat markings, and it was very inaccessible to humans. Starting in August through now, we have continued our scat collection, and we were actually able to locate more active latrines along Caldwell Creek, which is the dot over by Titusville, Cussawego Meanders, and private property right outside of Meadville.
So moving on to our actual scat collection and storage. So when we're collecting these scat, we're looking specifically for fresh scat, which is characterized by a distinct fishy scent. They're typically more wet, so they're more fresh, and typically have like visible fish scales within the scat. So that's usually within 5 days of excretion. We're also collecting the soil that is underneath or near the scat sample, including some leaf litter, to try and detect some of the microbes detected in those samples. We're using specifically sterile 50 milliliter tubes and swabs so as to not contaminate our samples, and those are labeled with the date, the location, and the content of the samples. And storing the samples, we are storing those samples in a freezer until further lab analysis later on.
So the lab analysis of samples will be done in late November, early December, and we will be doing PCR analysis of the 16S rRNA gene. This will help us to separate otter DNA from the bacterial DNA, and we will be sending the bacterial DNA off to an outside lab to identify the specific bacterium. Our results, so as of now, we have collected 10 different pairs of soil and scat samples. Our most active latrine was at the Erie National Wildlife Refuge. We've actually been monitoring this latrine for several years now, and we've got as many as 6 different river otters on camera traps there at the same time. And we use camera trapping images at this latrine and at the State Game Lands 270 latrine to help provide concrete evidence on freshness. So in this picture, which the otter on the left is leaving us a nice fresh sample, and we were actually able to collect this about 10 hours after it was left, so it was very fresh.
So we're anticipating similar results to the former literature, which had a high diversity of microbiomes in the river otters that, or in the otters that were sampled in previous studies. So we anticipate that to be a product of the local diversity and the soil microbiome within our region. So more specifically, a lot of our samples are coming from the French Creek area, which is a highly biodiverse habitat with more types of fish and mollusks found in that area than any other stream in Pennsylvania. So this bar chart demonstrates the distribution of the gut microbiome within sea otters, which had primary food sources of crabs, mussels, and urchins. And we anticipate, while the sea otter study determined that their results were very distinct from any other otter studies that have been done before, we're anticipating that those results come out very similarly compared to previous sea otter studies with a very high diversity of microbes detected in their samples.
And we would just like to say thank you to the Watershed Conservation Research Center on Allegheny College for providing us with a mini grant to do this study. We would also like to thank the RSC Center at Allegheny College that promotes undergraduate research, the French Creek Valley Conservancy, the Pennsylvania Game Commission, and John Totten for letting us use their properties to collect river otter scat. Does anyone have any questions?
So other than using the game cameras, what are other ways of determining like the freshness of the scat?
So fresh river otter scat is very dark in color. It's usually like a dark brown or black. It's wet. And within the first week of excreting the scat, it will actually have a very like fishy smell, like that smell you get when you walk into a fish market, which is very strong. So it's really easy to tell that way if it's fresh or not.
But to get like the accurate like 10-day-old, you would use the game?
Yes.
Okay.
Yeah. In the back.
I'm curious what you hope to learn from the sequencing of the soils. Once you get back to an estimation of what the diversity of microorganisms is within, how do you intend to use that information?
So we're looking to compare like the gut microbiome of the river otter, like that diversity with the diversity of the microbiome of the soil, which will also help to show us how healthy that ecosystem is and if there's any pollutants and stuff like that.
What was the last?
Oh, and any like pollutants or pathogenic bacteria found.
What else is helpful with the comparison? What else will the comparison help you tell you?
Yeah, I think it's going to show us to what extent the soil microbiome is affecting the gut microbe composition within those river otters, because we know that it does have an effect just from previous literature, but I think more specifically within our region, within our area, it's going to be helpful to show, like Libby had mentioned, the health of the surrounding environment and then how that contributes to the gut microbiome composition of the river otters in the area.
Have you caught on film any otters actually consuming the soil?
Oh, no, not at our sites anyways. But the prey that they eat lives, like the salamanders and frogs and stuff, yeah.
Yeah, so when we say that the soil microbiome composition is affecting the like wildlife gut microbiome composition, they're not necessarily like consuming the soil directly or anything. It's just their exposure to the microbes that are present within the soil. Like the previous study showed, the sea otters that they were sampling weren't consuming the soil there, but it was just an accumulation of the microbes that were present in the soil, as well as the prey that they were eating that were exposed to the microbes in the soil as well.
Any other questions? All right, thank you. And I think our last presenter is here, is Bryce. Do you have your flash drive?
I just have it on my computer. Is that not okay?
I might be able to make, no?
Transfer it by a flash drive.
Yeah, we have a flash drive up here. Do you have a, yeah, if you can just bring that up here, I can try to get it transferred over. While we're doing that, if you guys want to grab refreshments or anything. That's okay. can set your laptop down on that table if you want. All right, our last presentation for this session is characterization of bacteria from wild mushrooms in Allegheny National Forest by Bryce Hansen.
It's just this button right here.
That's for the laser, and then you can use these to advance or go
Okay,
back on your slides.
Hello, my name is Bryce Hansen, and I'm an environmental science and sustainability student at Mercyhurst University. I worked on this study with my partner Hannah Bozak, and we worked in Dr. Mohan's lab at Mercyhurst University. Dr. Mohan is our microbiology professor. And in this study, we were investigating bacteria in wild mushrooms in Allegheny National Forest. So I love mushrooms. I think my love for mushrooms really began when I began to become passionate about the outdoors and the environment. And for me, this started during COVID-19. I'm from Wyoming, from rural Wyoming, and so my only option during COVID was really to go outside. And as I explored the outdoors, I just became really interested in these curious little creatures popping up out of every nook and cranny in so many different forms, colors, shapes, sizes, and in like the most precarious positions. And so when Dr. Mohan invited me to come on this hike with him and do this study with him about bacteria and mushrooms, I just could not say no. But before I get to the study, I wanted to give everyone here a little bit of background about what mushrooms are and why they're important to us and the environment. So Webster defines a mushroom as an above ground, fleshy, fruiting body of a fungus. So it's important for me to tell you that above ground can mean coming straight out of the soil, but it could also mean coming out of a tree. As you can see with that fungus, the jelly fungus, it's coming out of the side of a tree. So as for the taxonomy, I don't want everyone to get intimidated by all of the big taxonomy words or anything like that. It's just important that you know that mushrooms, all mushrooms are fungus, but not all fungus can create mushrooms. So that's the big thing here. And then there's 6 main types of mushrooms that you'll find in the wild when you're trying to forage for mushrooms. And these are the bolete mushrooms, gilled mushrooms, puffball mushrooms, polypore mushrooms, coral mushrooms, and jelly mushrooms. And actually all 6 are found in Pennsylvania, and we found 4 out of the 6 for this specific study. So now that you know what mushrooms are, let's understand why they're important. So mushrooms are important for us for nutritional purposes. For their caloric density, mushrooms provide an extraordinary amount of antioxidants and protein. For medicinal, they also have crazy cool medicinal properties. They've been used in ancient cultures for medicine. If you think of chaga tea, for example, where they dry mushrooms, dried chaga mushrooms, crush them up and put them into a tea for immune support. But then they're also being looked at for modern medicine. Certain properties of mushrooms that are extracted are thought to be anti-carcinogenic. So they could have implications for now and into the future. And then they're important for their ecosystems. Mushrooms have what's called the mycorrhizal network, and my PowerPoint kind of messed that up, but it's supposed to say mycorrhizal network. And this is the underground part of the fungus, and it stretches out from the mushroom. And science has actually proven that this network connects plants, connects roots underground. So it's possible that that this underground network of fungus is connecting different trees. So say one tree is struggling to have enough phosphorus, another tree that might have an abundance of phosphorus is able to trade through this network that occurs underground, and mushrooms all have a part in that. And then the other ecosystem service they provide is as a decomposer. And that's pretty much what you learned about mushrooms in the 5th grade. That they take something that died and then they turn it into nutrients that are available for new plants. So how are mushrooms able to perform all of these things for us? And that's why it's important to understand the inner workings of mushrooms, which is a widely understudied field. But if we understand the inner workings of a mushroom, we can understand how they reproduce. We can understand why they are so nutritious and why they have so many medicinal properties. And we can also understand how they exist in and have an impact on the environment. And so one of also the most understudied things in the inner workings of mushrooms is the role that bacteria plays. So bacteria inside of mushrooms, very little is known. A lot is known about bacteria that exists on mushrooms or in the substrate that mushrooms grow in, but very little is known about bacteria actually existing within the mushroom. And a lot of what is known is only speculation as to why that bacteria is there and what functions it might be providing for mushrooms. But from what is known, there are both mutualistic and parasitic or pathogenic relationships between bacteria and mushrooms. So the mutualistic relationships, bacteria are known to provide sugars for mushrooms to facilitate growth. And for parasitic relationships, bacteria are known to cause common blotch diseases that disrupt the cultivating industry. So with all of that being known, let's get into our study. So this started with Dr. Mohan inviting me on a hike with his nursing students. And Dr. Mohan had invited me on previous hikes, and he constantly noticed me veering off from the group and fangirling over all these different mushrooms that I would find off the trail. And he would be like, "Bryce, where are you?" And I was like, "Oh, don't worry, I'm over here just taking a picture, no worries." And so when he wanted to do this study, he thought that maybe my expertise in foraging would help him. And so we hiked the Allegheny National Forest Rimrock Trail, and as you can see, we got lucky because it was super overcast and rainy, which isn't ideal for hiking, but it is ideal for finding mushrooms, because if it's in a darker environment, then these bright, vibrant mushrooms really poke out at you. And as you can see, we found some incredibly unique mushrooms in our study. We were able to find 11 different mushrooms, and they were from four of the 6 different groups that I talked about. So also my formatting got messed up here, but it's called found polypore mushrooms, guild mushrooms, bolete mushrooms, and coral mushrooms. And so once we foraged these mushrooms, we had a set of procedures to get them into the lab and to start our experiment. So first, we tried to touch them as little as possible and put them in plastic bags. Once we got them into the lab, we surface sterilized the mushrooms to try to get all of the bacteria off of the outside, so we knew that we were only working with the bacteria inside of the mushrooms. Then after that, we ground up these mushrooms and diluted them into 3 different concentrations to try to see if they even had bacteria that would grow. And then we inoculated trays and tried to isolate bacteria. So once we had successfully isolated our bacteria, we had a couple lab objectives and approaches, and I included a picture. This is Hannah, my lab partner. She's not here with me today, but she ran a lot of these experiments once we got into the lab, so it's really important that I highlight her work. But first, we were trying to identify the bacteria inside of these mushrooms, and we did that through serial dilution plating and 16 RNA sequencing. And our second objective was to figure out if the mushroom bacteria were causing disease. So are the bacteria that we find in here, are they pathogenic or are they mutualistic? And we just did that through a quick infection assay. Then we tried to find what nutrients the mushroom bacteria like to feed on. So what are they doing inside of these mushrooms? And we did that through running several different biochemical tests, and we found really cool results for sugar, citrate, and phosphate. And then our last one, we tried to find the different growth conditions that the mushroom bacteria could tolerate. So we tried to grow these mushroom bacteria at varying temperatures, all the way from freezing to super hot. And so what we found, the bacteria that we found inside of here, Ewingella americana and Pseudomonas species were the most common species that we found inside of the mushrooms. And for the Ewingella americana, this was the most prevalent type inside of the mushrooms. And from research, it is shown that Ewingella americana can cause blotch disease on mushrooms, but they also facilitate growth, and they maybe inhibit the growth of mold on fungus. So they both perform good and bad relationships with mushrooms. As for the Pseudomonas species, this was the second most prevalent type found inside of our mushrooms. And they also are known to have both pathogenic and mutualistic. The pathogenic also being blotch disease, while the mutualistic relationships, they can protect the mushrooms from certain diseases that people have found in other studies. So with that being known, and with the bacteria that we're finding, we knew that they had both good relationships and bad relationships. So we wanted to see maybe what they were doing inside of our mushrooms. So we took our bacteria that we had isolated, and we took a handful of mushrooms from the cafeteria at our school, and we did an infection assay. And we inoculated these common portobello mushrooms with the different bacterial strains that we found to see if they were causing good things to happen, bad things to happen, and just kind of what the result would be. Now, it's important for you all to know that this study is very preliminary and not a ton of conclusions can be drawn just because these mushrooms are culinary mushrooms, whereas the bacteria that we originally got from were wild mushrooms. So it can have different relationships in different mushrooms. So that's important for you to know. But our study did show that only 2 out of all of the bacteria that we tested caused disease on the mushrooms. So this made us wonder, what are all these other bacteria doing inside of there? So that's when we started running these different biochemical tests. And the first biochemical test where we got a really interesting result was with citrate. And we guessed that the bacteria inside would probably be able to utilize citrate as an organic carbon source just because citrate is the second most abundant organic acid inside of mushrooms. And we were right. Over 80% of our bacteria can use citrate. And how this test works, the media starts out as a green color, and if the bacteria can use it, it turns into a blue color. So what this might mean for the mushrooms and for their mushrooms' roles in ecosystems, I found some interesting research that suggests it might mean that mushrooms participate in phytoremediation. Now, it is known that in Allegheny National Forest, there's high levels of heavy metals in the soil due to past industries moving through. And with high levels of heavy metals in the soil, it is known that plants secrete citric acid, and citric acid binds to those heavy metals, and then they are taken back up into the plants. And so once they're taken back up into the plants, the soil is alleviated. And so it's also known that mushrooms are shaped by the soil that they grow in. And so it's quite possible that there's an abundance of citric acid in the soil that these mushrooms were growing in. And so if they're the mushrooms are probably taking up all of the citric acid found in the soil. And so it led me to think, what if mushrooms are part of this process of phytoremediation as well? And what if they could be useful in the future if we're trying to clean up soil? So another biochemical test that we ran was for sugar. And we also thought that the bacteria in these found inside of our mushrooms could utilize sugar, just because sugar is one of the foundational parts of mushrooms. It's actually a building part of the cell wall of fungus. And we were right, a lot of the bacteria could utilize these different sugars. And so what might this mean? This might mean that the fungus and the bacteria are performing a mutualistic relationship. I found in a different study that mushrooms feed bacteria sugar that they have, and the bacteria turn it into something that the mushroom can then use. So maybe that's what these bacteria were doing inside of the mushroom. And this might be significant because people are using the sugars that they're finding in the cell walls of fungus to possibly combat cancer. So perhaps the bacteria inside of these mushrooms are developing these sugars that are anti-carcinogenic. So maybe the bacteria have a part to play in that. And then for our last biochemical test, we tested if the bacteria could solubilize phosphate. And a positive test is shown by a halo around the culture. So as you can see, a lot of our bacteria could solubilize the phosphate using secreted phosphatase, which is just something that breaks phosphate down. And so with that positive test, what could that mean? it also means mushrooms are known to produce phosphatase. And so if the mushrooms are producing phosphatase, what if we always thought that the mushrooms were producing phosphatase, but it was really the bacteria inside of these mushrooms that are actually producing it? And what might that mean for the mycorrhizal network, where trees and other plants are giving the fungus the products of their photosynthesis, and then the fungus are giving back to them different waters, minerals, and nutrients, and one of the main nutrients that is known is phosphate. What could that mean? Could that mean that the bacteria are the main role players in that? So then another interesting, all these results were expected, but then we found one result that we did not expect at all, and that was the growth conditions. Mushrooms typically grow between 12 degrees and 24 degrees Celsius, but for some reason, the bacteria that we found inside of the mushrooms, over 60% of the mushroom bacteria grew at 2 degrees Celsius, and over a quarter of the isolated species could grow at freezing temperatures. And so I have absolutely no idea why this could occur, but it's just very interesting. And what might this mean for when it is freezing outside? What are the bacteria that exist? What are they doing? And yeah, so that's obviously an area that needs explored further. So with all of this known, how is this important to us? Well, I think it's important because we can better understand how the bacteria inside of mushrooms are playing a part in the mushroom's nutritional value and in their medicinal value. It can also help us understand why mushrooms are so valuable for the environment and future directions that mushrooms might be able to help us, future ways that mushrooms might be able to help us enhance our environment. And then it also just helps us to better understand unique contributions and opportunities that come with understanding mushrooms and understanding the inner workings of mushrooms. So for the future directions of our study, we want to identify the remaining mushrooms. We didn't get to identify all of them, and so that's an important factor that's missing. We also want to rerun our preliminary study where we took the common portobello mushrooms, but we want to do that with wild mushrooms to see if the relationships are different with the bacteria in those wild mushrooms. And then we just want to continue to investigate our significant results. We want to investigate, are the mushroom bacteria really playing a principal part in the secreting phosphatase? Are they an important part of the citric acid cycle? All of these different things, just to continue to look in towards those. And so with all of that being said, I invite all of us to look at mushrooms and bacteria bacteria inside of mushrooms, much as we look at bacteria inside of humans. It was once thought that bacteria inside of humans only cause problems and cause diseases for us. However, new science is showing that bacteria is fundamental to our existence and to our health. And I would like for us to think of the same thing for mushrooms. A lot of people think, and the mushroom cultivating industry thinks, that bacteria just disrupts their functions. But what if bacteria performs life-giving services to these mushrooms? And then what does that mean for the mushrooms? And what does that mean for us? Thank you very much. And these are my references.
We have time for some questions, if anyone has any.
Yeah, go ahead.
Do you remember what the average number of bacterial isolates you were able to get per species happened to be?
Yeah, we found 2 to 5 mushroom isolates per mushroom.
Okay.
Yeah.
Did you happen to, or has anyone else in your group maybe sequenced the bacterial symbiotic community on the fungi to see what the non-cultural diversity is like? It's probably much greater than 2 to 5 that you're able to.
Yeah, I would guess so, but I'm honestly not sure. Like how I said, Hannah did a lot of the work in the lab, but we'll be back with our poster from 6 to 8 if you wanted to maybe look at that and ask her a few of those questions.
And lastly, did you mention what kind of media you use to culture your isolates?
We used, oh, I can't remember what we used. I'm sorry. I'm sure that she'll also have that on the poster. Thank you, though.
That, like toxicity or whatever, the infectious assay that you ran, would you like to do that again, but maybe on cultures that you grown to see, or on mushrooms that you've grown to see, like the effects on like a blank sort of soil sample rather than just, already like wilder, like, dead, culinary mushrooms and stuff?
Yeah. No, that would be interesting, like taking a soil that doesn't have maybe high levels of citric acid and seeing if the bacteria inside of the mushrooms could also digest citric acid. Was that kind of what you're saying?
I think so.
Yeah, I think that would be super interesting, just kind of seeing, and seeing how connected are the bacteria in the mushrooms to like where they are located. I think that would be super interesting.
Any other questions?
Yeah.
Just to follow up on your fungal identifications, were those sequence identifications or were those IDs just made by
No, we did sequence them.
the fungi?
Yeah.
So the few that you labeled as unknown?
Oh, I'm sorry. I thought you meant the bacteria. The fungi, we have not sequenced them yet. That's one of the things that we want to do in the future. Yeah.
All right, if there's no other questions, that concludes this session. Thank you, and
We're going to resume here now. So this is our last session of today. My name is Sarah Magian. I'm the Regional Science Consortium's aquatic biologist. I'm going to chair this one. So we have 4 presentations for our last session of oral talks today. Reminder that we do have our poster session and art exhibition this evening from 6 to 8 p.m. Very excited for that. We have our new highest number of posters we've ever had, so very excited for that. But first, we're going to get through this session. So our first presentation is Decomposition Markers in Standard Soil Analysis by Hannah Skropitz. What?
Okay, so today I'm actually presenting what I presented my thesis on, which is decomposition markers in standard soil analysis. Postmortem interval estimation aids investigators in reconstructing the events surrounding death and deposition and helps to narrow the list of potential identities of a decedent. Currently, there are different markers and techniques being used to estimate PMI based on the condition of the remains. Forensic pathology can be used within hours to 2 to 3 days. Forensic entomology can be used within days to a few months, looking at things like physiological temperature, which can estimate in days to weeks, and then ecological succession, which can estimate in weeks to a few months. But this method is currently pretty imprecise and largely in the experimental phase. And then also forensic anthropology, which is currently using a method that uses accumulated degree days to estimate PMI. Currently, the Megacy method is being used by forensic anthropologists to estimate PMI. This method uses visual scoring of decomposition and splits the body into 3 regions: the head and neck, the trunk, and the limbs. These area scores are added together to produce a total body score, or a TBS, which is then put into an equation to estimate accumulated degree days, also known as ADD. The average temperature from the previous days are added together, moving backwards from the day of recovery. This continues until the sum of the average daily temperature equals the estimated ADD from the equation. Some of the issues with this method include that it was only supposed to be a pilot study, but now it's being used as a full-blown method. The scoring is based on photographs alone. 68 of the individuals in the study were from 19 states, but 40% of these were from Indiana and Illinois. Greater than 75% of placement was during warmer months, which likely led to faster decomposition due to the warmer temperatures. And PMI estimations are less than or equal to a year, so we're not seeing the full range of decomposition process, and it's also not useful in fully skeletonized remains. Soil chemistry is currently being explored as an alternative method. Currently, there are a lot of soil chemistry testing methods being researched using multiple chemical analysis methods. Problems with these methods include that they have a pretty small sample size typically. They require equipment that is very expensive, and the testing can be time consuming. And also the labs that have these equipment to do the testing have a large backlog of casework because they are often forensics labs. There are studies looking at long-chain compounds like putrescine and cadaverine, which are mainly responsible for the smell that we associate with decomposition. But these methods require expensive equipment like a GCMS to test for these compounds. There are also simple molecule studies that are looking at things like pH, total carbon, nitrogen, potassium, etc. And while these compounds are easier to study, they've been proven not to be as reliable markers for decomposition. Researchers are developing methods using an electronic nose to develop an odor signature, but these methods also require expensive equipment, training on how to use this equipment, and the equipment is very weather dependent upon its function. Multiple studies have also looked at pH as a PMI indicator, but their results have varied. All results have shown an increase in soil pH, but the timing of this increase has not been agreed upon. Some studies saw a large increase in pH between day 15 and 36. Some saw an increase between day 14 and 23, and then a decrease at day 30, and then some saw a decrease after 1 year. Sampling designs of previous studies have widely used full bodies as specimens. Problems with this are that full body research is not as accessible. Results can be clouded by multiple processes happening during decomposition, and also the difficulty of having multiple replicas when using full bodies. Instead, tissue sample-based research provides easily accessible samples. Research can be conducted at a smaller lab facility. You have a much easier time of having multiple replicas, and also this method is much more cost-effective. So from all this research knowledge, I decided to look at which chemical markers are most promising for analysis of decomposition and that can also be tested in a cost-effective manner. I chose to look at compounds that you will see in standard soil analysis. Among those, I specifically focused on ammonia and pH based on the background research that shows these are stable compounds that remain detectable for long periods of time, and also due to their known presence in anaerobic conditions. And I specifically decided to focus on pH since there was quite a few discrepancies on the presence of pH in my background research. Moving on to the materials and methods. I used 56 soil samples that were collected over an 8-week span. These samples were removed daily for the first 8 days, and then every 3 days for the remainder of the experiment. When preparing the samples, first I weighed and labeled 100 milliliter empty beakers. Next, beakers were filled with roughly 80 milliliters or 4 tablespoons of soil that was not compacted down into the beaker. Tissue samples were then added, and the tissue samples I used was pork tenderloin, focusing on that muscle tissue. Then cheesecloth was placed over the beakers and secured with a rubber band. Beakers were placed in cardboard holding boxes that were labeled using a row column labeling system. Temperature was monitored and recorded using data loggers and taken every 5 seconds. And then I used 2 different types of data loggers to ensure temperature readings were consistent, and also one of the data loggers had a longer lifespan. So even though this was a rather simple experimental design, I was able to obtain very similar sample weights for the soil and the muscle tissue. Moving on to data collection. 5 samples were randomly selected each time for sampling. Cheesecloth was removed from the beakers. The beakers were then weighed with the tissue, the soil inside. Then the tissue samples were removed in the beaker with the soil was weighed, and then the soil was removed and just the empty beaker was weighed. Observations of the conditions of the tissue and the soil were recorded for each sample. After all weights were taken, 4 of the 5 samples were placed in a closed Ziploc bag and labeled with the appropriate information. And this is what a picture of one of my sample bags looked like. Originally, I had wanted to do the testing myself, but that proved not to be an option. So instead, I reached out to the Penn State Agricultural Analytical Services Laboratory to see if they could do my testing. After being approved, I submitted Ziploc bags to the lab for analysis. Analysis conducted at the laboratory included monitoring pH, soluble salts, total solids, volatile solids, total nitrogen and carbon, carbon to nitrogen ratio, and the amount of ammonia and sulfur present. So to analyze these results, I used correlation methods, linearities tests or runs tests, as well as curve fitting and least square regressions models. And I'll discuss these more in my results section. Moving on to the results. When determining where to send the soil samples for analysis, I found that each state has a soil testing service through a university. For Pennsylvania, Penn State offers these services, and this is the lab that I chose to use. These labs do testing that range from soil, water, and compost testing to plant analysis. Originally, I thought the best option was to submit my samples as standard soil fertility testing. But when I sent the first batch of samples to the lab, they informed me that the samples as constructed were actually compost samples. The requirement for compost testing was different than soil testing, but the lab was still able to test for everything that I had asked for. After receiving my results back, only pH and the concentrations of soluble salts, solids, ammonia, and sulfur correlated with time. All time-correlated compounds were most strongly correlated with the levels of ammonia. Indeed, the formation of ammonia and ammonia byproducts explains the concentration of the rest of these compounds, except for sulfur. but this can be explained by being one of the main byproducts of protein decomposition when with ammonia. The relationship of both of these variables was nonlinear. So I had to find the best fitting nonlinear model for this relationship. After trying different models, I selected the one-phase association model. This model is used to study kinetics, and in particular, kinetics of the interaction between the ligand and its receptor, or substrate and an enzyme. Ligand binding rates plateau when all receptors are occupied. This makes sense as why this model best fits my data, because we're observing the enzymatic degradation of proteins. Plus, we also expect saturation plateaus for both ammonia and pH. pH increases rapidly to plateau after approximately 2 weeks. Oops. So for pH, we saw a strong fit model that time explained 90% of this variability. So the rapid increase in early plateau would explain the discrepancies in previous studies because of the nonlinear relationship that we're seeing here. It should be noted that the rate of pH increase would be different in different soil types. So your rate of plateau depends on the initial soil pH and also the soil's buffering capabilities. For ammonia, the fit is similarly strong, but the plateau is much later, around 9 to 10 weeks. This shows that ammonia would be useful for longer PMIs than pH, and it could also be used to potentially useful marker for decomposition rates and to estimate the degree of decomposition. Moving on to the discussion. This study illustrates that basic standard chemical analyses show promise as a means to develop PMI estimation methods. They do not require complicated sample collection or preparation methods. They are affordable and available in all states. Methods are highly standardized, and the results are pretty easy to interpret. I was able to use the Penn State Agricultural Lab, and their submission guidelines were 1 quart of compost submitted in a Ziploc bag and labeled. Under the conditions of the study, the proportion of ammonia and pH showed to be highly promising to assess PMI. The relationship between time and both pH and ammonia contents were found to be nonlinear, which it helps explain the different rates of pH increase reported in other studies. The plateaus being reached at different times also provide a minimum and maximum PMI. For example, if we had a sample that had already reached the plateau for pH, but not the plateau for ammonia, you would know that the PMI estimation would be greater than 2 weeks, but less than 60 days. We also can generate a more precise equation. To generate this equation, we only have to analyze 2 factors, which are the two things that are changing in this model. Y0, which is your initial conditions, and K, which is your rate of change. First, you have to modify Y0, so your initial conditions, which could be your concentration of ammonia, or the initial pH of the soil, to see if K changes or remains constant. Then you can modify temperature, soil type, or the microbiome, etc., to see if your rate of change changes when these factors change. Once these models are tested and estimated, you can estimate the PMI prediction equations. A second element of note is that we can develop precise equations based on testing similar to larval rearing in the lab. These results open a wide array of potential studies based on this model, such as testing at different temperatures, testing in sterilized versus non-sterilized soil, testing in different soil types, testing the water retention and granulometry of the soil in anaerobic and aerobic conditions, and also testing with longer experimental times to monitor things like the pH decrease, the plateau of ammonia, and also the decrease of ammonia, and looking at other compound fluctuations that are at that longer time interval. There's also the potential to create a soil type database across the US, so we have a baseline for each level and what that pH would be. These are just some acknowledgments of who helped me as I was doing my research, some references. Do you have any questions?
Thank you. We do have time for questions.
It's also, yeah.
It looked like the full span of
Yes.
The soils and the little chunk of coral
Mm-hmm.
were basically kept at the same conditions, is that right?
Yeah, so they were in a lab at roughly like 70 degrees that whole time. So you also could do some sort of testing with different temperatures as a previous or other
I guess
study.
I was wondering about like climate, like if a cadaver was outside, you would rain within 60 days.
Yeah.
What effect do you think that might have on like pH?
I don't know what the rainwater would do to that, but definitely decomposition would change based on your weather conditions. How that would affect your soil, I don't think it would change it too drastically as you'd see like a big jump or a big decrease in pH, but I'm not certain. Thank you.
Any other questions? All right, thank you.
Thank you.
All right, and up next we have the use of anatomical variation, antemortem skeletal trauma, and pathology in human identification with Rhiannon Toy and Aurora Venna. There is a clicker up there if you want it.
All right, so we focused on the use of anatomical variation, antemortem skeletal trauma, and pathology in human identification. And just some background, this is a preliminary study, so we are just in the beginning phases, but we're studying the frequency of skeletal variation, antemortem trauma, and pathology in different individuals from our donated skeletal collection in the Applied Forensic Sciences Department at Mercyhurst University. And we selected 10 individuals at random from our selection to begin performing this data on. And some more like vocabulary background. So anatomical variation are structures that differ from those typically described in the majority of individuals in a given population. And then antemortem trauma is injuries sustained by an individual while they are still alive, hence antemortem, before death. Pathology is any signs of a disease present in the bones. It can be like more severe or less severe depending on the individual and how they react to the disease, how it affects the bones.
So we split up our data into cranial data and then post-cranial data. So I'm going to go over the cranial data first. When we were looking at the crania that we selected, we were looking for 3 different things. One was antemortem trauma, which would be any evidence of healed fractures or surgical devices such as plates, screws, implants, wires, anything like that. We are also looking for anatomical variations, such as a supraorbital notch instead of a foramen, which is seen in that very top picture. On the left, you can see that it's a notch. On the right, you can see that it's a foramen. We were also looking for more than one supraorbital foramen. You can slightly see above the notch in the first picture that there's a foramen right above it, and a foramen is just a hole in the bone. We were also looking for multiple infraorbital foramina, which is in the picture below that top one. You can see that there's that large one, and then more closer to the edge, to the corner of the orbit, you can see another one. We were also looking for the presence of a metopic suture. And the metopic suture is formed because initially when you're born, your frontal bones are actually in 2 pieces. And that metopic suture is a result of those fusing. So most of the time, you do not see that metopic suture because it does fade. But that picture, the second picture on the top, you can see that this individual has the metopic suture from their nasal up to their coronal suture. We were also looking for a number of mental foramina, which is seen in this very bottom one. You can see that large one, and then there's another one on top of it. And then we were also looking for the presence and number of worming bones, which are bones that are formed inside of sutures of the skulls. They're essentially extra bones. You can see that in that picture. The lambdoidal suture is coming down, but then there's also another part of the suture, and you can see that there's a bone formed inside of it. The last thing we were looking for was pathology, which included a number of dental restorations, such as amalgam filling, implants, or crowns. We were also looking for alveolar resorption, which is when your mandible and or maxilla are resorbed because you lose teeth. so that socket is then reabsorbed. We're also looking for periodontal recession, which is evident in the teeth when your gums recede and it tends to leave a line on the teeth. Now, in the 10 individuals we went over, only one showed evidence of antemortem trauma, and on their left parietal bone, you can see the indentation in their skull, which shows that it is a healed fracture. Then, out of the 6 cranial variations we checked for, the metopic suture and worming bones were the least frequent, thus they are more valuable to identification or more unique to a person. A later presentation today will go more so into the frequency of cranial variation specifically, but this is all the numbers that we got. It's divided up into if they had a notch, foramen, neither, or both, how many they had on both their right and left side for the various foramina, if they had a metopic suture, and if they had any worming bones. And then pathology. So periodontal recession was the most common pathology that we saw in the crania, because as 9 out of 10 individuals had it, 8 out of 10 individuals had alveolar resorption, and 1 individual had complete alveolar resorption of their maxilla and mandible. They had no alveolar sockets present, and their mandible and maxilla were severely reduced. For dental restorations, there were 7 out of 10 individuals. 1 had ceramic crowns, 5 had amalgam fillings, and 1 had implants and a crown bridge. You can see the crown bridge in the second one on the bottom in the implants. You can see the screws and then a bridge going over top. Then in that first one, you can see slightly the presence of broken implants in the very top. And then also in the back, you can see another forming of a bridge. 2 individuals had semi-erupted third molars, which means that their third molars had erupted through the alveolar bone, but they did not pierce through the gum. So they could not technically see their third molars, but it was in the socket. And the most individualistic pathology we found was mandibular and maxillary tori, which is this extra bony growth that you can see in this one picture. Only 1 individual had that.
All right, and then focusing on postcranial, which is everything other than the cranium and the mandible. So I focused on antemortem trauma, so I looked at healed fractures and surgical hardware as well, and some variations. The only one that really was glaring to us was the suprascapular foramen versus a notch. And then pathology, we looked at bony overgrowth and then ossified cartilage. All right, so an example of antemortem trauma, we found evidence of healed rib fractures in 3 out of the 10 individuals we looked at. And you can see from that picture that there's a callus forming and ossifying, and that tells you the step that this individual is in their healing process, because bone is a dynamic tissue, which means it repairs itself, and eventually that callus will thin out and reabsorb, and then it becomes the bone again. And the reason why there's a crack down the middle is because it was broken again post-mortem, but that's not forensically significant, while the antemortem trauma is. And then another piece of antemortem trauma we looked at was surgical hardware, which was also present in 3 out of the 10 individuals we looked at. And as you can see, it's very individualistic. There's surgical wires, and there's a lot of things implanted into it, and that can be good because you might be able to find serial numbers on it, and if they have surgeries and stuff done, they normally have really good antemortem records for comparison, which is really good for identification. And then for anatomical variation, we only found 1 individual with a very distinct anatomical variant, which was a suprascapular foramen rather than a notch. And these pictures were from the same individual. One was the right scapula and the other was the left scapula. And on the one scapula, you can see that there's extra ossification, so it's forming a foramen, a hole, instead of a notch, which is a lot more common. So as soon as we saw the foramen, we were wow, this is individualistic, this is cool. So that's very exciting. And then pathology, we looked at bony overgrowth, which was also present in 3 out of the 10 individuals we looked at. However, these were the more extreme cases. I think at least every single donor had some kind of bony overgrowth, just because it is very common to have like more ossification going on, like as you age and things like start to fuse together, because like as we stated, bone is a dynamic tissue and it's constantly growing and remodeling. And then significant ossified cartilage on 1 out of 10 individuals, and this was the example. We picked it up, and it was a very extreme case. Like as you can see, like the ribs are very fused to the sternum because of the cartilage becoming ossified, and then even the verts are all fused because of that ossification.
Now, in order to see the relevance of these antemortem trauma, cranial variation, and then pathology, we took a look at how the cases handled by the Applied Forensics Department at Mercyhurst were identified. We decided to take a look at 5 cases where dental, surgical devices, morphology, fingerprints, and tattoos were all used to identify the person. As we see, antemortem trauma, skeletal variation, and pathology were all used to positively identify someone. Now, from this we can draw that antemortem trauma is a lot more valuable when it comes to identification, as it's going to be more unique to the individual. It's going to be unique as to how it happened and then how it healed. And taking a look at these 2 rib fractures, one from earlier, you can see that the callus formed are very different compared to the 2.
Just like when we're talking about how it's unique when you're healing from a fracture, it depends on like what kind of fracture it is and like the health of the individual and other factors like lifestyle factors that might be involved, genetics. There's a lot of different things at play, so it makes sense that each fracture is going to heal a little bit differently. But what's important is as the bone like heals, it goes through different processes that like we can track. Like as you can see, like the callus, as I said, that's like earlier on in the process of healing versus the second image where you can see like the callus is beginning to like thin out. It's not as extreme, so it's beginning to like remodel more into like how the bone should look.
And that's all we have, so if you guys have any questions.
We have time for questions.
Yeah?
No, I'm sorry.
That's okay. I was just curious, that one individual that you showed, that 1 out of 10, it had like the ossifications and the fused rib cage.
Mm-hmm.
What would cause that? Is that like from some form of genetic thing or trauma? I mean, it just seems so bizarre.
Yeah. It could be like age, it could be genetic, it could be trauma. It just depends. We don't like have a lot of these, like we don't have information about the personal lives of these individuals, but based on how extreme it was probably a mixture of like different things.
Okay, thank you.
Can you talk about the collection? And is this an atypical collection, or where did this come from? Where did these individuals come from?
So these individuals, the donated collection, some people after they die, they will donate their remains to our university, and we will go through the process of skeletonizing these remains to put them in our collection so that undergraduate students and graduate students can use them as a study tool to further their education in the field of forensic anthropology. Some of the donated individuals may have come from cases that the department worked on, and either their loved ones or the coroner donated those remains to us. But we have individuals ranging from 2013 to 2023. The 10 we sampled for this project were from 2016 to 2023, but we hope to survey the entirety of our collection so that we can get a better estimate of what is more useful.
It's also a very diverse collection, so there's males, females, and then like different ancestries, which makes it very good when we're doing research, because then we can be more broad and we can really focus in on how different traits are different amongst different groups of individuals.
So how did you choose the 10 out of the whole collection? Was random or was it
It was
like,
random.
strategic? Okay.
It was random. I walked around our bone lab and I was like, we're gonna do this many from this year, this many from this year, and I just kind of went at random so that we could get hopefully a variety of different kinds of what we were looking for.
And then I have another question too, but
Mhm.
so when you don't have necessarily personal histories about these, do you still have like more basic information generally, such as the age of the individual, maybe cause of death for some of them, like stuff like that?
Most of them do in fact have their age, their sex, and then their ancestry. Some of them do have unknowns for that information. We'll also go a little more into that because we did have to look into that for the cranial variation one that we will talk about later today. Some of them just didn't have that information, unfortunately, but whatever information that we could find, we did take that into account.
Okay. Because I was also like, very interested in that ribcage result. I was like, like what's going on?
Yeah.
So, in your research, did you notice any differences between your male and female, people? Like in their, fractures and stuff like that?
Not really. It was kind of like we found more extreme cases of ossification, obviously, in like the older individuals. We didn't really notice anything when it came to males or females. But I would imagine that as we look at more individuals, that would become more apparent. Any other questions? Thank you.
Thank you, guys. Just gonna wait a quick couple minutes, like a 2-minute break. Just stay on schedule.
Yeah.
Alrighty, I'm gonna move along here. So our next presentation is The Importance of Comprehensive Dental Records and Identification Efforts by Joe Atsirias-Garriga. Thank you.
I'm always impressed how you say my last names.
Thank you.
Okay, so, well, what I was going to talk today is the work product of three people. Hannah Skrovitz, that you have already met her, and also Rupina is a high school student, which is impressive. So she has a lot of interest in forensic odontology. So we are tutoring her in a research project that is like, that's what she is doing. So, well, first of all, I would like to talk about forensic odontology. Who are the forensic odontologists? Normally, forensic odontologists are not very popular as forensic anthropologists because we never appear in Bones or CSI or anywhere. So we are dentists that can deal with cases of dental age estimation. Actually, dental age estimation is a very hot topic for the research and the cases, especially for instance, like unaccompanied minors in some countries. So it's very different if they are like over 18 or below 18 years old of age. So, and after that, like, of course, like every case that we have no idea about, like those remains, who they are, the first thing that we have to do is to reconstruct a biological profile, which is like a broad description of the individual, female, male, what was the age, approximately like in a range, like it was like a male individual that was in between 30 and 60 years old, and the stature is another item of the biological profile, along with the ancestry whenever we can state a population affinity. But forensic odontology, it doesn't contribute at all or doesn't contribute so much for sex, well, actually at all, for sex estimation, doesn't contribute, of course, for stature, and we can have some hints in the dentition about the population affinity of the individual, but there's not so definitive. So the best contribution of forensic odontology in the biological profile would be the dental age estimation, and especially in subadults, so in juvenile individuals, which is like very, very, very accurate and very precise method of aging the juveniles. The personal identification, which is like once we have this biological profile, then we are going to be given by the authorities or by the law enforcement 2, 1, 3, 7. Sometimes I remember I had a case where I had 11 individuals that were the candidates for that identity, and then we have to compare the antemortem records with the postmortem records. Okay, so that's what we call personal identification. And if the rock stars in biological profile are the forensic anthropologists, the rock stars in personal identification are the forensic odontologists, because the means of like comparing the antemortem and the postmortem data, it's something that it's very fast, it's very reliable, and it's not that expensive. So also, forensic odontologists can deal with cases of human abuse and neglect, and that can be with the elderly, with children, any kind of case of physical abuse most of the times, and neglect. Part of these human abuse cases imply the bite marks, and the bite mark examination is part of it. It's a very controversial part of the forensic odontology realm. In some states, it's not even admissible as an evidence in court. And this, like, we know that we have to be very cautious. And actually, the American Board of Forensic Odontology has stated like how to issue the results in a very, very careful manner right now. So as for now, we cannot identify the biter that we used to do that in early times, but now we cannot exclude that biter, right? So what forensic ontologists have made is like, I cannot exclude that biter from having caused that bite or that bite mark, but we should rely on further investigation with the law enforcement and other evidence, which is very cool because we don't have that pressure on us, right? So, and also we can deal with civil litigation. So civil litigation, it's like, imagine that you go to the dentist and you go for third molar extraction, and that day your dentist is drunk or whatever, and he pulls up your central incisor. So then you sue the dentist. So that's what civil litigation is, okay? And this is an extreme case. I never had a case like that. So, but we had a lot of cases that are like, wow, right? So, as you can see, this can imply the living individuals. Civil litigation, it's, well, hopefully it's the living individual, right? Because that means that the dentist didn't kill the person. And also some dental age estimation and some bite mark analysis, we do them in alive individuals. So as for like, if you compare the types of cases that we have in forensic anthropology, particularly all of them are analyzing the remains, the decedent, right? But in forensic ontology, we can have some cases that imply the living as well. Personally, I nearly all the time I deal with the decedents rather than the living. But every once in a while, you have a case of a bite mark that the kids are still alive, or the female is still alive, or whatever. So as I was saying, so that the personal identification is what is like the core component of forensic ontology. And that's why, for instance, we are always present in all mass disasters. So we are so needed for comparing the postmortem and the antemortem data. According to Interpol, we have three main identifiers. So those primary identifiers are fingerprints, odontology, and DNA. And those are like, a primary identifier would be like, I just need one of these methods to scientifically identify someone. If I have already those results from DNA, I don't need to do forensic odontology. I don't need to do fingerprints because I already have the DNA. And the other way around. So if I have fingerprints, why do you want to spend your time and your resources doing odontology or DNA? And odontology, it's, yeah, if you have odontology, you're good to go. You don't need fingerprints, you don't need DNA. So why I'm talking all the time about like, yeah, so identifying the individual is so cool by the dentition. It's because our dentition, our dental tissues will preserve throughout all the phases of decomposition. So, and actually they will resist a lot of the environmental factors. And so many times we can see that any environmental factor will affect first the bones and after will affect the dentition. So maybe fire and some chemicals can damage, of course, the dental tissues, but they will affect first the soft tissues, of course, and the bones, and after they will affect the teeth. And we have not only just the teeth itself, we have like all the dental work that we perform in our patients. And what I always say is that dental treatment, it's like haute couture, it's not pret-a-porter. So haute couture, you're like you know, like Christian Dior or Gucci, that they made like these special dresses for that person, and they count every millimeter of their, that's how we perform dental treatment. It's especially for each patient. We don't have fillings that are size XS, S, or M. We don't have that, right? So it's especially for each patient. It's performed for every patient. And And we have, apart from the teeth itself and the dental tissues, we have all the treatment, but we also have all the maxillofacial structures that are around the teeth, that they are like very good references, anatomical references. And so that makes that just looking at an oral cavity, we have a lot of features to pay attention on that can have an individuality. So we can have a filling, we can have a curvature in a root, we can have like a trabecular pattern that can be like very distinctive. So we have a lot of information in just like in a pretty small space of our body. So that makes a very accurate, very reliable scientific means of identification, and also it's really fast. So it's really fast once we have the antemortem records. When we have the antemortem records and we can compare them with the remains that we have, this is so fast. The tricky part is to find them, which is not our job. It's like the job of the law enforcement, and they do that brilliantly. And as I said, like it has like minimum cost for medical examiners and coroner's office. I always say like the only time that you will get a cheap dentist is once you die, and it's so true because forensic odontology, it's really not a great expense at all. And we don't need like very sophisticated reagents or very sophisticated machinery, right? So what we have is in any identification process by dental means, we'll have our post-mortem data collection. So that will be everything that we are going to take, the information that we are going to take from our remains, like every photograph, radiographs, and those are like mandatory to have. And also all the notes that we'll have, the odontograms, which is like the dental inventory. And there in our department, we know that we are very picky for how to take the radiographs, how to take the photographs. We're very picky on that, right? So you will have all the information of the remains, and maybe you don't have the antemortem yet. So you're waiting for the law enforcement to find it and bring it to you, right? So at that stage, we will take all the radiographs, we will take all the photographs, and there will be certain things that will pay our attention. Like, do I have fillings? You're like, wow, bingo. Do I have a crown? Wow, that looks so good, right? But actually, until you don't get the antemortem, maybe that's useless, right? So if you're lucky, then you get the antemortem records, and then that's when you compare, okay? And the way to compare the antemortem records and the postmortem records, it will be drastic differently according to what antemortem records did you get. If I get like, and let me show you what we are comparing, right? So we are comparing where the fillings are, where and how the material looks like, and it's all based on the radiopacity, how radiodense or how radiopaque does the structures look like in the radiograph, right? So we can see that these two here, it's a molar, and we have like this filling here, and we see like same radiodensity, so probably the same material and same shape. Okay, that's a match. And we have like this one here that has like this same shape, this same material. It's a match as well, okay? And we have this guy. I really like this one because like the shape, it's kind of like unusual. So the more unusual that it is, something that it is, it's the more that we like it because it's more individualizing, right? And we see that here, so that will be a match. So all these three, that will be a match, and a match. So that really looks good. Also, this guy here, it's present and it's not restored and looks exactly like this. So it's another match. Then we can have what we call explainable discrepancies. So we have everything matches, but what happened here? Well, maybe that individual got a filling in a later date of this radiograph. Maybe was not even in the same town. Maybe not even like maybe he went to or she went to Brazil and had a toothache and had to put a filling there, right? So an explainable discrepancy is everything that could have happened between the time gap between like when when the dentist took the radiograph and until I find the remains. And then, okay, we can have explainable discrepancy until a point. So there are things that cannot happen. So we cannot have an antemortem filling and then have a healthy tooth. We wish, right? But this is not possible. So that will be an explainable discrepancy, and according to the combination of much explainable discrepancies and explainable discrepancies, we can have a positive identification, or we can have an exclusion, and we can have something in between that, well, the dental records do not depict very well, so I cannot give a very good comparison. So I really don't know if that's this person or not. Most of the times, when we say it's a positive identification, or it's we have insufficient dental evidence, most of the literature says that it's because of the type of antemortem dental records, right? So we can have these antemortem dental records, which are not very good, and we can have like more fancy ones like those ones, right? Also, the postmortem the postmortem condition of the remains, that would be so important, because like if we have like some crowns and some roots that are separate because they have been burned, and and there's some of them that I would not be even certain which type of tooth it is. Okay. So we wanted to see like more like what in our department, what type of dental records we have. Okay. In our sample, we look at 80 cases from 2019 and 2024, and those years are because we incorporated forensic odontology in 2019, and what we saw is that all these 100% of the cases used dental as a means of identification, and we had written dental records, we had radiographs, and we had CT scans. So what we saw, it's like a very, very, very dramatic difference when we just have written dental records, which is just like the dentist saying like filling in the occlusal, okay? Than if we see in an image that filling in the occlusal. So we see the shape. So whenever we can see the morphology, we can compare the morphology like antemortem image, postmortem image, okay? And also another thing that we saw is that the age of the decedent, it's very, very limiting, especially in the juveniles. You have less possibilities of having a radiograph, and also depending on the gap time that you have between the remains and the radiograph, that could be so changing. If we have the radiograph when that individual was 10 years old and then the remains are 30 years old, that changed a lot, and sometimes even not even all of the morphologies can be of help. Surgical devices were used in 2 of the cases, and that was so to corroborate the because if we have a surgical device, we will always make sure that that matches as well with the identity, right? So a limiting factor would be also like if the antemortem records are not available. We cannot do anything, like anything. The only thing that we can do is to upload all this information in the database of the missing and unidentified people and see if eventually at some point we will have a match, okay? But that's the only thing. So as we could see, like the times that we had dental radiographs, and so many times that the post-mortem condition of the remains were like very, very poor, there were so many cases that we could still use forensic odontology for identifying the individuals, and that's very good. So again, it's a primary identifier, and this is a fact. Maybe we should explore for other primary identifiers like surgical devices. I don't understand that why don't we have this as a primary identifier. And we get like minimal cost again and very fast results once we have the antemortem. So once we have the antemortem, sometimes we have called the authorities and say, look, is this person? Give me some hours to write the report, but it's this person. So you don't have to spend more resources to do fingerprints or to do DNA that are normally more, especially DNA is more expensive. So this is everything that I wanted to let you know, but again, this is not my work product only. It was Hannah and Rupina's work product that we are always amazed that a high schooler could have done that, right? So thank you so much. If you have any questions, feel free to ask them to Hannah.
Thank you.
Thank you.
Do we have any quick questions? We have time for like maybe 2.
This doesn't have anything to do with the research that you just, well, for someone to do this type of work, is it like a 4-year program or like?
Forensic odontology in me.
Yeah.
So forensic odontology, if you want to be called a forensic odontologist, you have to be a dentist.
Okay.
So you have to be a dentist, and then you are specialized in forensic odontology, right? But there are so many things that forensic anthropologists can do with the dentition, like dental age estimation. They are perfectly able to do that, and sometimes there are like some methods that have been developed by forensic anthropologists rather than forensic odontologists in age estimation. Also pathology and trauma, analysis of the skeletal trauma. Teeth are part of this of the skeleton. So normally forensic anthropologists are like, oh, teeth, oof, no, like they can. So we are not afraid to evaluate like multiple fractures, gunshots, the entrance, the exit. But you have a tooth that is chipped out, and they are like, oh my goodness, like yeah. So how we are teaching our students at Mercyhurst in forensic anthropology, it's as if they were dentists. They won't be doing dental identifications by themselves, but everything else, forensic dental age estimation, pathology or trauma analysis in the oral cavity, they are like more than prepared for doing that.
So, do you ever use stable isotope analysis in teeth?
We can do that, and actually in teeth will give us like very old data. So when we are like, in our childhood. So if you have it like in bones, that will be in a recent date, and you have it from the hair, that will be like very much recent date. So actually in teeth, you go to the childhood of that individual. It's really good, because the teeth do not remodel. So when they form, they're done. And so that's why if you chip one of your teeth, do not expect to form a callus or anything. So you will have the chip, and you will have to go to dentist and put some material in it.
All right, thank you very much.
Thank you.
All right, and for our last presentation of this session, we have quantifying skeletal variations in the human skeletal collections of the Applied Forensic Sciences at Mercyhurst University. And is this once again Rhiannon Toyna Roravana? Okay, come up.
So this research was also done with another undergrad student at Mercyhurst in the Forensic Anthropology program, Michaela Brinkley. Unfortunately, she was supposed to give this presentation, but she is not feeling well, so we're stepping in for her. And we're just going to get started.
All right, just so you have some background, we selected 25 individuals at random, again, I know, of the Applied Forensic Sciences Department at Mercyhurst University to analyze the frequency of 6 cranial variations. So we looked at the supraorbital foramen versus a notch. We looked at the number of supraorbital foramina, the number of infraorbital foramina, the number of mental foramina, and the presence of the metopic suture, and the presence and number of wormian bones. And out of the 25 individuals, 6 were female, 14 were male, 5 were of unknown sex, 3 were black, 16 were white, and 6 were of unknown ancestry. So it's a very diverse selection. And the individuals also had a diverse age range from 42 to 90. And 42 is pretty young considering the other ranges that we have. 1 was in their 40s, 4 were in their 50s, 10 were in their 60s, so that's a really common age range. 1 was in their 70s, 2 were in their 80s, and 1 was in their 90s, and then 6 were of unknown age.
This is just illustrating kind of what we've already talked about, just the variations that we were looking for. You have the overall picture and then zoomed in, just kind of showing what we were specifically looking for.
This is like frequency distributions of the different traits. So it's a box plot that shows that the metopic suture, as well as like the wormian bones and the mental foramina, were like the least frequent, which makes them like the most valuable for like identifications, while the other ones were more frequent. But there's also a lot of variation involved, which is why we need a more diverse and like large sample.
Now, I broke this up by doing, when looking at most frequent cranial variations, I broke it up by overall, which was the supraorbital notch instead of a foramen. Then by our age groups, as we only had one individual in the 40s, 70s, and 90s range, I did not include that. When we had multiple individuals, I did go and look at what the most frequent variation was. For the 50s age group, it was the supraorbital notch again. The 60s had a tie between supraorbital notch and then multiple supraorbital features, so whether it was multiple notches, multiple foramina. And then in the 80s, the supraorbital notch was also the most frequent. By sex, females had supraorbital notch, multiple supraorbital and infraorbital foraminas were the most frequent, and then males had supraorbital notch as the most frequent. By ancestry, those of white ancestry had the supraorbital notch as the most frequent, and then those of black ancestry had the supraorbital notch, multiple supraorbital and infraorbital foraminas. And then going by least frequent cranial variations, overall the least frequent was the multiple mental foramina. And then going by age once again, in the 50s the metopic suture was the least frequent. In the 60s, the number and presence of wormian bones was least frequent. In the 80s, there was a number of these. There was multiple supraorbital and infraorbital features, metopic suture, and then wormian bones. For females, the metopic suture was once again the least frequent. In males, the presence and number of wormian bones was also the least frequent. And then by ancestry, those of white ancestry had metopic suture and wormian bones as the least frequent. And then those of black ancestry had wormian bones as the least frequent. However, individually, these variations don't give us much information. That's why we have to look at the most and least frequent combinations of these features in order to get valuable information for identification purposes. Now, overall, the combination of supraorbital notch, multiple supraorbital and infraorbital features was the most frequent variation combination that we saw. By age, those in their 50s tended to have the supraorbital notch, multiple supraorbital and infraorbital features as a combination. Those in their 60s had the supraorbital notch, multiple supraorbital and infraorbital features, and multiple mental foraminas as the most frequent combination. In the 80s, it was really just the supraorbital notch was the most common between the individuals that we had. For our female donors, we found that the supraorbital notch, multiple supraorbital and infraorbital features were the most frequent variation combinations. And for the male, it was supraorbital notch, or the same thing, I'm sorry. And then by ancestry, those of white ancestry tended to have the supraorbital notch and multiple supraorbital and infraorbital features, and then same thing for those of black ancestry. And then this is the most important data that we got out of this, as this is the least frequent combinations. So this is what is going to be most valuable to human identification. Overall, the presence of both a metopic suture and wormian bones was the least frequent cranial combination. By age, those in their 50s, some of them had all 6 variations, but that was very few, so that was the least frequent. Same with the 60s. Those in their 80s had multiple superorbital and infraorbital features, and then wormy bones as the least common combination. And then by sex, our female remains had all 6 variations as well. And then for the males, it was all of these, so the superorbital notch, multiple superorbital and infraorbital features, multiple mental foramina, and metopic sutures. So the only thing that the males did not have in their least common combination was the wormy bones. And then by ancestry, those with white ancestry, the superorbital notch and multiple superorbital and infraorbital features was the least common combination. And then those of black ancestry, the least common combination was metopic suture, multiple mental foramina, and wormy bones. Now, as multiple mental foramina, the presence of a metopic suture and the presence of wormy bones were the least frequent variations. They are the most valuable when it comes to identification, as they're going to be more individualistic to the person. And as the presence of a superorbital notch instead of a foramen and multiple superorbital foramina were the most frequent, they're less valuable when it comes to identification. This research was really just to find what is the least common and what is the most common in order to gauge which ones are better for human identification. As this is a pilot study, the research project led by Dr. Joe, which involves pattern recognition and comparison of cranial traits by artificial intelligence.
And like the more individualistic a trait, that information can be very useful to artificial intelligence that can then be used for identification. Because a lot of the methods we have are based on qualitative data instead of quantitative data. So the more quantitative data that we have, like the stronger like our identification can be.
Do we have any questions?
Yeah.
Why would there be a variation in these features between different age groups? Are these structures that would change throughout a person's life? Or is that just how data shook out?
This is how our data kind of shook out, but let me just go back. You're asking about the most frequent by
All
age,
of them.
essentially?
Like why would, because I'm not an ophthalmologist, I don't know too much about bones anyway, but why would some feature be key in someone's in their 50s versus women in their 60s? Does it just depend on the sample?
It has a lot to do with variation because we are very variable, which because humans are unique, that is how we can identify individuals. So these structures that we found that were more frequent might not be the case for every individual. It just depends. And as we expand our research, we might come to different conclusions that a different trait might actually be like more or less frequent. It just depends on the people that we're sampling and the kind of like factors that might be at play, which might be like genetics, pathology, and like age and stuff like that. It just depends on what is affecting the bones.
Can I jump in?
Yeah.
So this is like depicting like what were the results, but this individual that had superorbital notch on 80s, he had it when he had 20s.
Okay.
So this doesn't change like with the aging, but like that's like how they saw those results. It doesn't mean that we have an interpretation there that like only people that are 18 or older got like this. So the features will be like those features will be the same ones when we're adults but younger.
Very cool. Thank you.
Very related, and I don't mind whoever answers. But is it possible to have one of these features and have it disappear as you get older? Because I know you said like where like the, like say for example like where the skull heals together, you usually have, I forget the
The
proper.
metopic suture?
But yeah, it's exactly that. But can that actually, you said that could go away, right, in your other talk? You said that
Yes,
actually,
so
like,
most of the time the metopic suture is not going to be present as you get older because it normally your frontal bone, it fuses into whole when you're very young. So most of the time, you're not really going to have that metopic suture present, but some individuals do. We had one individual in our collection that had a very, very prominent one instead of just in the nasal area. It went all the way back to his coronal suture, which separates the frontal bone from your parietal bones. So it's not a very common variation, which is why it was very valuable for us to look and see which skulls had the metopic suture and how far it went.
Okay, because then like some of these features, could they change over time, or are these all ones that wouldn't, I guess?
These will be like all the same when you're an adult.
Okay.
When you're a kid, no.
Okay.
But when you're like achieving like, a teenager, that will be it.
Thank you. I have no idea what I'm talking about. Any other questions?
So this is for identification purposes. What's like the baseline at which they would be compared to? Because like I don't, what would be person? Is there like not really dental records in terms of like a database of what skull features someone has living usually, or are there?
That's also a question for Dr. Joe, as this definitely relates to her project.
Like how we're using those cranial features to identify the individuals rather than the identity.
Sure, yeah. And I guess when you have a skull, how would you like see who that is if that person didn't have those features reported when they were skull? Sure. Yeah. Yes. Yeah.
Yeah. So we can have like a CT scan.
Okay.
From a skull. So normally the more frequent ones are like cranial. Well, the more frequent ones are the panoramics. But not we have cranial CT scans a lot, and we have like cases where we identify like very ridiculous traits. And with the CT scans, the ratings that we can build on, we can do a 3D reconstruction that we can build on, so we can even have like cases where we took a picture, we scanned with a surface scanner, and then we took the abdomen CT scan, and we can see the same profile or the same trait or whatever. And then also chest radiographs, sometimes they are given, and knee, and then hip radiographs. It's hospital records.
Okay. I see.
Good question. I was kind of wondering the same thing. Any other questions? All right. Thank you, guys. All right. And that was the last of our oral presentations for today. We do have oral presentations going on for the rest of the week, so Thursday and Friday. Basically all day, both days, so please come back and feel free to attend those. But then also tonight at 6 p.m., we have our poster session. There's a poster in here. Oh, there's two. Sorry. I didn't notice yours. But so please do attend our poster session. We also have an art exhibition this year. So that is a new addition. So we will have, I believe, right around, I should know this because I've registered everyone, but we have like right around like 15 to 20 artists, and we have many art pieces coming tonight. So that will be happening at the same time as the poster session. So it'll be busy and exciting.
So art going to be upstairs, or is it all going to be down below?
It's all right down here. So actually where the registration desk is, you know how that kind of curves around? That's right where the art is. It's like basically right at the end of our registration. And then all of the areas where posters will be, you'll see the numbers for the posters on those boards. We do use the same boards, so it's a little confusing because the art will be hung on those same gray boards. But they'll also be displayed on tables and TVs and all that kind of stuff. So, yeah, please come. All right, and then we can break. Thank you, guys.